So very warm welcome to everybody. A very good morning to all. So this is the fourth day of this RC program, and it's a great opportunity for me to introduce and first of all welcome to our resource person for today's session, Professor Madhu ji Tapadia ma'am. Welcome ma'am again from our behalf.
Thank you.
So thank you ma'am, thank you. So let me give a brief introduction of our resource person today for all the audiences. Professor Madhu Tapadia is a senior faculty member in the Department of Zoology at Banaras Hindu University, where she has been teaching and conducting research for more than 2 decades. She completed her education and PhD at BHU and later worked as a postdoctoral fellow at the National Institute of Immunology, New Delhi. Her research focuses on understanding how organs develop and function using Drosophila as a model organism. She has contributed important insights into how certain cells avoid cell death during development, how fruit fly kidneys can help us understand human kidney related disorders, and how genetic changes influence conditions like stress, depression, and neurodegenerative diseases, including Huntington's and Alzheimer's disease. Professor Tapadia has published widely in national and international journals, contributed chapters to scientific books, and guided several research projects funded by agencies like DST and SER. She also holds leadership roles such as Executive Secretary of the Indian Society of Cell Biology and is a member of the Indian Society of Developmental Biologists. With a strong academic background, dedicated mentorship, and impactful contributions to developmental biology and neuroscience research, Professor Tapadia is widely respected in the scientific community and continues to inspire students and young researchers across the country. So, with this brief introduction, now I will hand over my microphone to Professor Tapadia. Ma'am, the stage is yours now. Thank you.
Thank you so much. First of all, for inviting me to present this, and second, to all the participants who are online. Uh, well, I would like to say that when this topic, I was thinking on what to talk. So, this topic, which was suggested, and I took it as a challenge because
Site for
which I took as a challenge because actually my work does not directly relate with nanoparticles or nanotechnology, nanoscience, but I work with the renal system, which I'll talk about very briefly. And but however, when I went through the literature, I found very interesting, and what I could relate was that these nanoparticles, very small particles, if you think about our ancient medicine, so where we used to have bhasmas, so these bhasma, it may not be exactly the same as nanoparticles, because in bhasma you have varied shape, but then there are some particles which are actually same as, or they have a similar size as the nanoparticles. So since I work with some of Ayurvedic formulations, so it was interesting for me to understand how bhasmas can be related to nanoscience, nanotechnology, and if we can somehow, do more intensive research on these bhasmas, so probably we can also use them for different ailments. So, so I'll just start. I'll just share my screen. Is it visible?
Yes,
Yes,
ma'am,
ma'am.
it is.
Okay. So, so I have tried to gather some information about nanoparticles and renal diseases, and then we'll be talking about its benefits, drawbacks, and then later on I will also talk about how we can study these nanoparticles in different model systems. And the model system that I will be talking about is Drosophila. Okay, so the flow of the talk will be basically what are nanoparticles, and then how Drosophila helps as a model system. And in Drosophila, specifically the renal system of fly, which we have been working on very intensively since last several decades. And then and then before that, I'll talk about nanoparticles in renal diseases. So, okay, so before I start, if you have any questions or if you don't understand anything, please let me know. And if you feel that I am, there's some facts that I have not mentioned correctly, then also please, I will be happy if you can correct me. So, as nanotechnology is very uh, it's it's the upcoming science these days, because we all are talking about the nanoscience, nanomedicine, nanotechnology, and so on. So what exactly does nano means? So as the name suggests, nano is, it comes from a Greek word which means very small. Okay. And the range is 1 to 100 nanometer. And 1 nanometer, if you it's difficult to imagine, but one nanometer is actually 1 billionth of a meter. So here there's increasing times. Are you able to see my pointer?
Yes, madam.
Okay.
Yes.
Yes, ma'am.
Okay, so you can see this is increasing time scale over here, and if we start from the widest, the biggest time, the biggest scale that is over here, that's 100 micrometer. So it's a human hair, which is 100 micrometer, and then as you go down, the scale decreases, and we have at 1 nanometer there are carbon nanotubes, and then before that there's carbon quantum dots. And so these carbon nanotubes, quantum dots, so these have actually revolutionized the entire therapeutic approach for many diseases. Okay? Now the history of nanotechnology. So though we are more into nanotechnology now, but actually it started long back, around 2000 years, when the sulfide nanocrystals, they were used by Greeks and Romans to dye their hair. And then this image that you see over here is actually the glass painting, in which there were gold particles of different sizes. And what here I would like to mention is that when we reduce the size of any element, okay, then at different size, they absorb different colors and they reflect different colors. So they acquire the different color when they go from macro to a large size to a nano size. So this is what was used, these gold particles. They were of different sizes, and because of this different sizes, they could reflect different light, and so therefore they were used to paint windows. And the scientist who was actually in 1959, you would have heard of this scientist, he's a very famous physicist, and he's Dr. Richard P. Feynman. So he talked about there's plenty of room at the bottom. What exactly he meant by there's plenty of room at the bottom? That means, if it actually means that you can you can compress lot of information in a very nano size at the bottom. So plenty of room. It's like, if you have a all pin, so the head is broad, but as you go down the tip, there's a very fine tip over there, which means that uh, which means that you can encompass many things. You can encompass many things. Are you able to see?
No, ma'am.
No, madam.
Ma'am, you just share your slides again. It will be visible, I think. Select the entire screen, ma'am. I think, ma'am, you select the entire screen. You just again go to the presentation icon. Yeah.
The presentation. So
Then
present
present
something
sharing.
else. It
Then
says something.
you share the entire
Entire
screen.
screen.
I think it will be visible then.
Ah,
Yeah.
okay, okay.
Yeah. Yeah.
No, no.
No, no, no.
Ma'am, you can go to the PowerPoint slides, then it will be visible.
Yeah. Wait. But I'm not. Is it
Ma'am?
visible now?
Yes.
Yes,
Yes,
ma'am.
ma'am. Yes.
Yes, ma'am.
Yes.
Yes.
So I'm also learning, online presentation is a challenge for me actually. So yeah, so we talk. So there's plenty of room at the bottom. That means you can encompass lot of things in a very small tip. So this is how the science of nanotechnology grew. However, the term nanotechnology was first used by Norino Taniguchi in 1974, and then IBM took it up and they developed the scanning tunneling microscope. And now we know that in 1991, the first carbon nanotubes were discovered. And after that, it has actually, it has, it has grown so much that now we have really fine, very, very fine nanoparticles which are doing, and there's lot of science that can be done with this. So, actually, nanoscience is when you can transform, or uh, you can develop very micro level particles from very bulk materials. And one thing is very interesting, that when a particle is its original bulk size, the properties are very different. But when it is reduced to nano scale, the properties change. And actually this ability, this change from the large size to a nano size actually removes the toxicity that it would have had. So and then it makes it more favorable to use. Okay, so and this alteration is more useful for biology. So nanotechnology, it's a science to create new materials from the already existing materials, but reducing them to a nanoscale. And a nanoscale is typically 1 to 100 nanometers. And this, just I got this very interesting uh, information, that the human nails, it grows one second. So human nail, it grows 1 nanometer every second, and hair of beard of males, they grow 5 nanometer every second. So you can calculate the extent of growth if anyone is interested. Then nanoscience, actually when it started, it has permeated into all fields of science. So it's actually nanoscience is very interdisciplinary, because there's physics, chemistry, biology, environment, economy, computer science, engineering, medicine. So there is no field left where the nanomaterials or nanoscience is not used. And they have, they are we have lot of advantage of using nanoscience, that we'll be specifically talking about its use in medicine. And just to give you some information, that these nanomaterials is present in almost all the products that we are using, right from clothing to food to medicine to cosmetics. So the spread of nanoscience is huge. And therefore, it is also very important to understand whether always it is useful. Are there any side effects, or are there any drawbacks, or advantage, or also there are disadvantages of these nanomaterials in our daily life? It's very essential to assess the disadvantages before we take it before we use the advantage of nanomaterials. So and in India is 19% of the Indian market, you know, is will has nanomaterials in some product or the other. And by 2030, it's predicted it will be 19%. Now, the unique properties of nanomaterials, actually the importance over here is, and this is a physics involved, that if you break any larger uh, particle into smaller particle, what increases is the surface area. So the surface area over volume is very, very important. So if the surface area increases, it gives us a lot of handle to either load drugs or put some kind of fluorescence, attach some kind of fluorescence, different kind of fluorescence molecules to it, so that you can also image where these materials are going. And in this image that you can see over here, the effect, you can see different colors. So these different colors are because of the size of different nanomaterials. And then the other unique property are the enhanced mechanical strength, and low weight. So they are very light weighted. So therefore, even so, it's not very difficult uh, for these materials to flow into the, if they are injected into the bloodstream, so they can flow very easily. And because of the enhanced tensile strength, the chances of breakage of these nanomaterials is very less. So therefore, therefore these are some of the properties which make nanomaterials useful for many different kind of applications. Another thing is high permeability. Now, what this high permeability essentially means is, in there are certain disease conditions in which the blood vessels, there's leaky blood vessels, so the especially in cancers and all, so the blood vessels is leaky. So therefore, because of this leaky blood vessel, the its advantage, the nanomaterials can easily enter through the blood brain barrier. And therefore, there are, because one of the main hurdles for transferring any drug into brain is a brain blood barrier. So these nanoparticles, because of the high permeability, they can very easily enter the blood brain barrier, and therefore you can deliver the drug very easily. Now, many countries, countries and many industries are using these nanomaterials for lot of use for consumers. Now, there are two ways to achieve the nanoparticles. One is called as top down, and the other is bottom up. So, as the name suggests, top down, bottom up approach is used in many different areas of science. And what exactly it means, top down is you have a larger material on the top and you manipulate it so that you get nanoscale structures. Or from bottom up is from, as it's shown over here, from the atoms to clusters and to nanoscales. So these are the two approaches by which one can develop nanostructures. And they the important point over here is that these nanostructures, nanoscale structures, they have much superior properties compared to the starting material, either the bulk or the atoms. Okay? So what are the different types? They are categorized first on one of the ways of categorization of nanomaterials is the dimensionality. So 0D, that means there is no dimension. So therefore they are quantum dots or fullerenes, which or gold particles, which are circular molecules. So so they are called as 0D. 1D is nanotubes. That means it grows in one direction. So you have carbon nanotubes, you have metallic nanotubes, and so on. 2D is when they grow in two direction. So you have carbon coated nanoplates. So these are plate or like spreads which are used for different reasons. And 3D is they are larger molecules in which they are spherical or they are cubical. That means they have some space inside where whatever material you want to transfer can go inside very easily. And nanomaterials, they are also categorized on the basis of the properties, whether they are organic or inorganic. So here I have shown some, this image shows some of the inorganic nanoparticles. So the carbon based, which is one of the very widely used element for making nanoparticles, is carbon. So you have carbon tubes, you have graphene, fluorite, then you have silica and polystyrene also. So they are inorganic. Similarly, quantum dots. You can see over here, these are different colors that have been given. And this is because the property of the quantum dot changes depending upon the size. And then the other kind of inorganic, you have gold, zinc, iron, silver. Now, the choice of nanomaterials, these materials, actually depends upon what kind of experiment you are trying to do. So a researcher actually decides which is the best for its use. And then you have gold nanoshells, magnetic carbon nanotubes, and so on. And in organic, you have nucleic acid based hydrogels and protein based ferritin. So you have these liposomes, micelles. So these are all organic. And as I said, depending upon the kind of experiment that you want to do, you can use different nanomaterials. Now, this is a repetition, I think, in which, one second, in which it has shown the application of nanotechnology in the area of producing energy, medicine, nanobiotechnology, nano devices, optical energy, defense security, bioengineering, cosmetics, and nanofabrics. And as I said, that this was the slide where I showed that nanomaterials has permeated into almost all areas of our use, immediate use, or the environment. Now, in cancer, this is in cancer, we know what's cancer, and uh, nanomaterials are very useful in treating cancer. Some of them have also entered clinical trials, and one of the reasons is because we can have targeted delivery of these nanomaterials so that they can go and they can enter into the cancerous cell rather than the other cells. One of the reasons could be that there are certain surface molecules in a cancer cell which we can make a nanoparticle carrying the drug to recognize that. And once it is recognized, then it can go inside, and the drug can be transferred directly. But of course, before using any of these drugs for cancer, it is very important first to check them in a normal cell. So even if by chance there's some kind of by chance if these drugs they enter normal cell, they should not they should not cause any damage. So all these kinds of checks have to be made before they are used for cancer. And so different kinds of drugs that are used, different kind of nanomaterials. You have quantum dots, and then you have supermagnetic iron oxide particles. They act as high performance contrast agents for MRI. See, it is also very important that we should, the spread of cancer. We know that cancer spreads very quickly, and when it spreads, that's a malignant stage, and or the metastatic stage. So at that stage, it's very difficult to actually find out which regions are under which cancer cells are spread to which regions. So these SPIONs, these can help, because if you add a fluorescent molecule to this particular nanomaterial, it will go and it will bind to the cancer cells. So you can actually have an entire image of the spread of the disease, and then one can decide what kind of treatment has to be given. Then you have surface functionalization, which allows there's an area to volume ratio, and so therefore it's very the amount of drug that can be put into that this nanomaterial increases. And then these are targeted delivery. You have over you so these are passive, active, and there are certain smart carriers which release cargo only in the response to tumor micro environment. As I said, that the tumors, they have acidic pH, or they have some elevated enzymes. So these facts are recognized by the nanomaterial, and so there's targeted delivery, which is very important, and that is one of the places where which is actually people are working upon, so that the normal cells in many other diseases also is does not take place. Now, so what are the advantage of studying nanoscience? It provides great surface area, less toxic, targeted drug delivery. They can enter through blood-brain barriers. They provide good retention. This is also very important because there are certain nanoparticles, nano carriers, which slowly release the drug. So this is important because the more the drug stays in the body, the more useful it is for that patient. So they're biocompatible, and then the controlled release of drugs and low doses can be effective. And then there are certain disadvantages, that this is not cost effective. Okay, preparing is maybe easy, but then its characterization is difficult, is costly, and then it requires sophisticated technology. Again, bulk production is limited till now. We don't have certain ways by which you can produce lot of drugs of nano size, and then the specific skills are required, and leakage of content is possible, which, as I was telling, that these nanotubes or nanocarriers, carbon tubes, so if they leak the content at wrong places, then that can be detrimental. Now we'll just talk about one this acute kidney function injury, AKI. So AKI is a condition in which the AKI is a condition in which kidneys that is acute kidney injury, in which there's a rapid decline of the kidney function, and it has so the number of patients of AKI is very high, and one of the main causes of acute kidney injury is sepsis, renal hypofusion, and nephrotic drugs. And there are a lot of advances in curing AKI, and there are certain, if you can see over here, that these are the nano carriers that are used in AKI. One is liposome. So liposomes are biocompatible, and they can carry both hydrophilic as well as hydrophobic drugs, and they stay longer in the bloodstream. And the important point about liposome is that their surface over here, it can be modified so that these liposomes can go and target in the kidney only. So therefore, delivering different whatever medicine is inside is very helpful, especially if you want to deliver some RNA, antisense RNA, or some anti-inflammatory drugs. So liposomes are very useful. Then you have this mesoporous silica nanoparticles. So they have large surface area, and the important thing over here is that the pore size in this the pore size is adjustable. That means you can either increase or decrease the pore size depending upon the drug that you are using. And again, you can also target it to go and bind to the damaged kidneys and deliver these antioxidants or whatever drugs is being loaded into them. Then there is this PAMAM, where they are tree-like nanocarriers that accumulate in the kidney. Then you have chitosan nanoparticles, which are biodegradable and then increase uh, cellular uptake, uh, cellular uptake, improve stability and solubility of the drug. Then there are DNA nanostructures. DNA nanostructures like TFNA, they naturally accumulate in the kidney and they can easily enter cells. They carry drugs, they improve solubility, and they act as therapeutic agents by reducing oxidative stress. Then there are exosomes. So exosomes are natural vesicles that are excellent. They have excellent biocompatibility with transport proteins, lipids, and stem cell derived exosomes reduce inflammation and oxidative stress. Now, there are different strategies to improve the efficiency of nano medicine delivery. One is the physiochemical properties of nanoparticles. So you have the GFB, that's the glomerular filtration barrier. They become leaky during AKI, and therefore the different particles can enter easily. As I said about blood vessels in the blood-brain barrier, if they become leaky, the nanoparticles can enter very easily. Then you can also modify the surface by adding certain ligands, which help in targeting these nanovesicles into the kidney, the required region. And then there are so they are applied for antioxidant therapy, for ferroptosis therapy, and anti-inflammatory therapy, autophagic therapy, and so on. So these are some of the ways by which one can use the materials to for ameliorating the damage of acute kidney injury. Now, I'll just come to another aspect, and that is the malphizian tubules of Drosophila melanogaster. So, what are these malphizian tubules? As I was telling that we need to have certain models to understand or to do certain experiments, because all experiments cannot be done on human. Cell lines are very good, but cell lines are cell lines because they do not represent the exact situation that is taking place in any organism. So therefore it is very important to have certain model organisms to understand or to study any sign, any aspect where you can extrapolate your data from one model organism to human. So, Drosophila, there are organs which are called as malphizian tubules, and these malphizian tubules, they actually function as kidneys in these insects. I hope you all are familiar with Drosophila melanogaster. This is a fruit fly, and it was first used in genetics experiments by Thomas Hunt Morgan, and since then it has become one of the most widely used and most favored model organism to study. So the main function of these malphizian tubules, they are its excretion, but apart from it, it also has immune response. So so this diagram actually shows similarity between kidney and the malphizian tubules. Now what you see over here is that in human kidney, the tubule region, the Henle's loop region is similar to the malphizian tubules in Drosophila, and the podocytes or the glomerulus region over here is similar to the Garland cells, which are also called as nephrocytes. So, as in kidney, you have a composite structure where you have the glomerulus and the Henle's loop or the nephron together. In Drosophila, it's delinked. So you have this region which represents the podocytes, and this region malphizian tubules, which represents the tubule or the nephron. Yeah, so it's clearly shown over here that the nephron, this region is represented by the malphizian tubules, and these are the Garland cells over here. Now, these malphizian tubules were first identified by scientists Italian scientist. He was a physician, Marcello Malfighi, and he was the first to describe. He was the first to describe many different scientific breakthroughs over here. You can see. So, and his diagrams of chick embryos, hand drawings, were so such with such detail that it will be surprising that what he drew just by simple microscope and or eye observation is almost same as we have not made any change actually in what he had made and what we see now. And this over here shows you the different malpighian tubules that he drew of different insects, and these are all hand drawings, and this is one, and with very precise sketches, and this is one art that is now diminishing, and especially in the age of AI now, I think it will get completely obliterated from the history. But these are something which we should appreciate. Uh, the the what human eye can do is it is very satisfying actually. Now, Drosophila is it completes its life cycle in four different stages. You have it's a holometabolous insect, so you have embryo, larva, pupa, and adult. And one of the important hormones for allowing all these transition from one stage to another is ecdysone. Now, I'll just talk about Drosophila melanogaster specifically because I work using Drosophila melanogaster. So, this Drosophila melanogaster has two pairs of tubules. One is the anterior pair, and the other is the posterior pair. Okay? So, there are many aspects, but what is very interesting, and if you have interest, we can also discuss later, that there are two main cell types of these malpighian tubules. One is called as the principal cells, and the other is called as the stellate cells. And you can see over here, using immunostaining, we have identified the different cell types. The red is the principal cells, and the green is the stellate cells. An important point over here is that as Drosophila melanogaster is a holometabolous insect, which means that from the larva there is complete transformation, metamorphosis into adult. And during this metamorphosis at the pupal stage, all the larval tissues are histolized, but malpighian tubules are one of the tissues, organs, which are not histolized during larval pupa, and they're carried over to the adult. So this with this question, I started my research, why malpighian tubules are not histolized. And then we have certain answers also. But what I also want to say over here is that these principal cells and the stellate cells, the number of these cells is invariant. That means once they are formed after embryogenesis, the numbers do not change. They remain the same in larva, in pupa, and in adult. Another important point you can see over here is that the precise way by which these cells are arranged in the malpighian tubules. These stellate cells are actually mesodermal in origin. You can see over here, and once they come, once they are mesodermal in origin, and they intercalate between these tubule cells, the principal cells. So the principal cells forms like this. It elongates to form a tubule, and then the stellate cells, the mesenchymal cells, they undergo a mesenchymal to epithelial transition, and they enter or they are intercalated within this tubule. So I think this is a kind of repetition where I want to just show that this is the nephron of human where you have these podocytes, and in the nephrocytes also you have these slit diaphragms. So they are kind of filtration unit in Drosophila. Now, it is very advantageous to use Drosophila because you can identify the different regions depending upon the function. There are certain ways by which you can target whatever gene or whatever protein you are interested in at specific regions. So in the Malpighian tubules, you have an initial segment. So there are certain ways by which you can express any gene only in this region. Then you have this main segment, which is actually the fluid secretion region, and there are certain genes or certain ways by which you can express your genes only in this region. And here is the lower tubule. So you can see that all these different colors in these regions are because of the different functional differences. And also you have ways by which you can identify only, or you can express any gene only in the principal cells and in the stellate cells. And here I would just like to tell that the name of stellate cells is because of its star shape. Because of its shape, the name of stellate cells, it derives its name from there. Now, these Malpighian tubules can help us to understand development, function, and or they can be used as a model to understand development, function, and immune response. How development? So this was my student, and what he showed, first student, and what he showed was, if you see over here, this is a normal wild type Malpighian tubule, and you can see these small are the stellate cells which are arranged, and these large ones are the principal cells. In certain conditions, here we have removed ecdysone from here, and what we see is that this arrangement is lost. Okay, the shape of the tubule also changes, and this arrangement is lost. You can see clustering of these stellate cells. So normally these stellate cells are spread almost at equal length throughout the tubule, but in the absence of ecdysone, this is missing. Also, one can use the larval mouth hooks for identifying the age of these larvae. So this is the third instar, and in mutants you can see, so the wild type has uh, proceeded to third instar, and in mutants they are still first or second instar. So you can study the development by using the mouth hooks, or you can look at the Malpighian tubules. Yeah. And then see, in the absence of ecdysone, if you can see this kind of change or in arrangement, and this is a rescue experiment where we added ecdysone receptor and we could see it rescued, and so you can see again the regular shape of the Malpighian tubule as well as the arrangement of the different cells. So I'll just skip this. Okay. Now, this is an important part of the study which is going on. And as I told you, that these Malpighian tubules, they do not undergo apoptosis. So initially we thought that probably they do not express the apoptotic proteins, and therefore they do not undergo apoptosis. But we did studies, and then 2 3 students they studied, and they found that it is not the absence of apoptotic proteins. The caspases are expressed in the Malpighian tubules. So the question became more intriguing as to, if these caspases are present, then what are they doing over there? Why these cells, these organs, they are not undergoing apoptosis? Okay, and uh, this here you can see this is a wild type Malpighian tubule, and in the absence of in caspase mutant. So the Drosophila caspase 3 is called as DRICE. So in DRICE mutant, it's very clearly seen that the tubule shape has changed. It acquires a cystic phenotype, and you can see it's in the cartoon pencil representation over here, sketch representation, how the shape of these Malpighian tubules change. And what we see is, when these are the embryos, and you can see that the normal Malpighian tubules at the later stage, they extend in the anterior and posterior side, and in the mutant, what you can see over here is that though the cells are present, but they do not acquire the shape. So, and that therefore the caspase, what we conclude is that caspase is important for the development of Malpighian tubules. So there are some mutants that we have identified which change the shape of the malpighian tubules, and one is the SERCA mutant, which SERCA is sarcoendoplasmic reticulum calcium transporter, which transports calcium from the cytosol into the endoplasmic reticulum. And in these mutants also, we see these. There is a difference in the malpighian tubules, and also Yorkie. Okay, I'll just skip this. Okay. So these mutants, one is Sarka and the other is Yorkie. Yorkie is from the hippo pathway. And what we observe is when these two molecules, the Sarka and the Yorkie, they are absent, the malpighian tubule length increases dramatically. So this is actually a side observation from other experiments, but I just thought I'll show you that how, because we're talking about development, so how different mutants can change the shape of the malpighian tubules, and this can be very easily studied. Also, one can use SEM to understand the surface morphology. So this is the wild type where you can see very uniform diameter and the surface topology is very there are not much changes. Their surface looks almost in all throughout the tubule. The surface morpho undulations remain same, whereas in Rice mutant, you can see the size, the difference in the size of these two different mutant and the wild type. And what you can see is we noticed there are several holes over here. So these holes have a consequence and it affects the function of the malpighian tubules. And this is again another student. What she did was she was looking into tau is basically, we know it's connected associated with tauopathies. Okay, and they are mostly, they have been studied for their expression or their role in neurons. But what she did was she looked into the malpighian tubules and studied the malpighian tube. She downregulated or took mutated the tau in malpighian tubules, and as you can see that there is gross morphological changes in the malpighian tubules when there is tau mutant, and it's very clearly seen over here that there are certain holes which are actually present. So what I want to say over here is that though tau has been studied in neurons specifically, but even in non-neuronal tissue, it has an important role, and she's trying to find out what exactly tau is doing. Okay. Now, in continuation with the development part, I told you about the stellate cells, which are mesodermal in origin. Now, it's very interesting that in the larva, this stellate cell voidal, and as it matures into an adult, at the pupal stage, it acquires the characteristic stellate shape. Okay. Now, in Dry's mutant, this is a wild type stellate cells, and this is in Dry's mutant, the larval stellate cell. So you can very easily see that these larval stellate cells in Dry's mutant actually resemble the adult, suggesting that dries, the role of dries, and importantly in this connection, that you can study mutants which affect the shape of the malpighian tubules, shape of the stellate cells, and you can identify what are the different genes that are involved in regulating the cell shape, because cell shape for almost all signs is connected with the function. So if there is a change in the cell shape, the function will be consequently changed. Now, how also you can study is by looking at the actin organization. This is, we have done, you can do phalloidin staining, and you can see these actin filaments, how they are arranged in wild type, and you look at the mutants, it has grossly changed. So this also gives one of the measure to understand what kind of change has taken place. And this is also same, but this is in different colors. So we have used different fluorescent molecules over here, and in function. Now, the function of malpighian tubules, it secretes fluid, and then there are certain particles which are present over here. These are crystals, we call it uric acid crystals, and these crystals in wild type, they do not accumulate to such great extent, so they constantly are made and they are flown, and then they are secreted out. But in these mutants, so this is specifically in the dries mutant, you can see that the huge amount of uric acid crystals that is deposited. Okay, and in one of the rescue experiments, we showed that these crystals, it's again like wild type. So you can do these kinds of experiments. So one student also looked at the crystal morphology by SEM, and you can see that the crystals are very regularly cuboidal shaped or rectangular, cuboidal or shaped over here. But in mutant, they appear amorphous. The crystal nature is actually lost, and this is because of change in the elementals percentages, and so this changes the crystal structure. And you can also see over here that this is the lumen. The size of the lumen in mutant is very huge compared to wild type. So all these luminal defects, they all add to the loss of functionality. Another way by which you can study is doing a functional test by using rhodamin 123. So normally rhodamin 123, if you incubate larvae, malpighian tubules in rhodamin, it's a fluorescent dye. If you incubate in larvae, incubate malpighian tubules in this fluorescent dye, then normally it gets flushed out from the system, suggesting that they are functioning properly. However, in mutant conditions, they are not flushed out, again representing that the function in these mutants are affected. Similarly, this is a Dry's mutant. You can see all these crystals that are deposited. And there's also disruption in the ion transporters. And one of the ion transporter is DRIP. DRIP is present in crystal cells and it transports chloride. So in wild type, you can see this green color, that's DRIP, but in mutant, it's not there. Another transporter, a very important transporter for kidney function, is sodium potassiumase, and you can see in wild type, you can see the color of the. This is immunostaining, where we use fluorescent dye to detect sodium potassium ATPase, and you can see that you have red fluorescence, but in mutant you don't see, suggesting that the expression of these transporter is reduced in these mutants, and so therefore it's affecting the function. Then immune response is very interesting aspect of these malpighian tubules. Insects, as we know, they only have innate immune response, and in the innate immune response also they have the cellular response and the humoral response. So the cellular response is by these mechanisms: phagocytosis, encapsulation, coagulation. And in humoral response, there are this antimicrobial peptide should come over here. So the antimicrobial peptides are released, which kill the pathogen. So there are two pathways by which the antimicrobial peptide is synthesized. One is a tall and the empty pathway. I'll not talk in detail about these pathways, just to let you know. Now, what we did was very interesting. This was the E. coli killing assay, in which what we did, we used, explain to you this slide, and don't worry about what's written over here. This is E. coli killing assay. In these experiments, what one does is one grows a lawn of bacteria on agar plate, and then you put certain antimicrobial peptide. So here what we have done is we put secropin, which is commercially obtained, and we have put lysate of the malpighian tubule, this pupal malpighian tubule. And what you can see is that there is clearing of the region. That means these the antimicrobial peptide in the malfetin tubule, they are capable of killing bacterial cell. And here, what you see is that we have fed these large uh, fluorescent bacteria, and you can see this was just an experiment to show that yes, they are actually feeding bacteria, because in one of the reviewers asked, how do you know that these larvae are feeding on the bacteria? So we did use this fluorescent GFP labeled bacterial cells, and we can see that they are present in the gut, suggesting that yes, they are feeding bacteria. And then this was we can see that there are these antimalarial peptides. They are expressed during different developmental stages. So one way to check by using LAGZ. So we have this antimicrobial peptide which is labeled with LAGZ reporter gene, and here this is labeled with GFP. So what I want to tell you over here is that you can use many ways by which you can identify the expression. So here you can see these are control. The top panel in each are the control, where you see less expression of these antimicrobial peptides. And when you treat them with LPS, there's enhanced expression of these peptides, suggesting that this LPS is able to increase or enhance the expression of these AMPs. These are again different ways by which one can study. This is by doing RT-PCR of different antimicrobial peptides. After this is after normal and then control and normal and so on. So you can see the enhanced expression and do a lot of studies like this. And then this slide shows this is relish. Relish is it's an NF kappa B transcription factor. So this NF kappa B transcription factor, when it enters into the nucleus, it is able to synthesize. It is able to increase the expression of AMPs. So normally, these are present in the cytoplasm, but when upon infection, they enter into the nucleus. And what we have shown over here, that if we increase the expression of AMPs and the survival of the larvae on the bacterial food increases. And these suggest that these larvae are able to dwarf off the effect of these pathogens. Important point here is that all these, there are 7 different kinds of AMPs present in Drosophila. There are different groups and they're all present. And what we see is they do not follow all or none. That means the expression of these AMPs at control condition varies at different developmental stages. So at some stage, some AMP is more, other is less, and so on. So it's not that either all will express or all will not express. What I think is the organism makes sure that some AMPs is present all the time, so that the organism is protected.
Ma'am, I want to just interrupt you a bit.
Yeah.
Navonita Ghosh ma'am has written that does gut dysbiosis in Drosophila alter its innate immune response?
Does gut?
Gut dysbiosis in Drosophila.
Yeah. So it alters the immune response. Yes, it does.
Okay,
That's a
thank you.
different line of study, but yes, there are some studies which are going on related to the gut dysbiosis, and that increases the expression of AMPs.
Thank you, ma'am.
And let me tell you over here that these malpighian tubules, okay, the main function is excretion, okay, but they also have this additional function of as an immune organ. They're autonomous immune organ. The main immune organ of Drosophila, they are the fat bodies. Okay, so in fat bodies under normal condition, AMPs do not express. So AMPs are only expressed when there is a pathogenic challenge. However, malpighian tubules are the organs in which we see basal level of expression. Okay, and this is what, see, as I told you, as the we start our work actually revolves around two questions. One is how these tubules evade apoptosis during metamorphosis, and second is why these tubules are not histolized. What is the significance? So, what I think is that during metamorphosis at the pupal stage, when the entire larvae is covered by the cuticle, the thick pupal case, so at that time the organisms, the pathogens which are entrapped into the pupa, probably they are taken care by the AMPs expressed by the malpighian tubules. So this is so therefore the significance of malpighian tubules, it is the first organ to respond to any pathogen challenge. So if there is gut dysbiosis, then you will see a systemic response. So I don't know whether the AMP's expression increases in the malpighian tubules, but the fat bodies, the AMP expression increases because that is the primary organ for synthesizing the antimicrobial peptides. Okay. Yes, so this is another interesting case where, as I told you, we have the cellular immune response. One of the cells is the crystal cells, and these crystal cells they secrete this phenol oxidase enzyme, which uh, which leads to melanization and killing of the pathogen. So you can see over here that at certain conditions, number of crystal cells is much more compared to wild type, and these crystal cells can be very easily visualized. You just have to place the larvae at 65 degrees centigrade. So if any one of you is working with larval Drosophila, and if you want to test whether certain conditions increases the number of crystal cells, what you just have to do is you have to feed these larvae on that particular uh, food containing any toxic substance that you are testing, and just keep them at 65 degrees centigrade, and you can count the number of these crystal cells over here. So there are different conditions. You can see that the number of crystal cells have increased, suggesting that yes, whatever compound you are testing has a uh, toxic property which increases the immune response. Now, why Drosophila? So Drosophila is useful to study many human diseases because as of now 75% of the different human diseases, they have their homology in Drosophila. If you so therefore it becomes a very important model to understand, study different diseases causes and all. And even if a particular gene is not expressed in Drosophila, so Drosophila offers a very unique advantage where you can express any human gene at in a tissue specific or a cell specific manner. So therefore Drosophila is, I find it a very useful model organism, and with other properties that we all know how Drosophila is useful. Now, some experiments that we have done, I'll just show you. We'll just talk about rice mutant. So what you see over here is in rice mutant compared to wild type, there's a change in share. There's huge change. Okay. So one of the pathways that we looked was the hippo pathway. Another pathway was the TOR pathway, and then next the other pathway is the calcium signaling. I've shown you some images of hippo and circa, but what I'll show you over here is that in rice mutant, the expression of all of these increases. So this is, you can see, this is uh, immunostaining with Yorkie. Yorkie is a transcription factor. You can see enhanced expression of Yorkie, enhanced expression of MEC, enhanced expression of SERCA. And when we do rescue experiments, that means when we reduce the level of Yorkie, reduce the level of MIC, and reduce the level of SERCA, you can see that the malpighian tubules, they regain their shape, and the expression of these different transcription factors are same as wild type. And this is the tubular phenotype which you can see. And you can also see the actin organization. If you remember, I had shown you in rice mutant there was huge gray, very enhanced actin levels, and the organization was disrupted. But now they all come back to same as wild type. And here, if you see, TOR is the MIC is the transcription factor, and it is by the TOR pathway. So what we did here is we treated with rapamycin, and we could show that there is rescue, suggesting that yes, TOR pathway is involved. Okay. And as I was telling you about the Ayurvedic formulations, so we have been using Ayurvedic formulation for in one. Initially, we used there are two Ayurvedic formulations: the Guduchi, that is the Giloy, and the Madhu. Okay, so they are both known immunomodulators, and we you worked on, or one some students are working on the Huntington disease using Drosophila as a model organism. And in this Huntington disease, uh, when we simulate Huntington disease in Drosophila and feed them in Guduchi and Madhu, so instead of finding cure, what we observed was it actually aggravated the disease. That means the degeneration was much more. So in that paper, we concluded that it is not always that Ayurvedic formulations are always good. So under normally it is good, but under certain disease condition, they should not be taken randomly, and therefore very lot of precaution or caution has to be taken, because there are a lot of information, these days in social media about holistic medicine and Ayurveda and all different kinds of herbal drugs. It may be good, I'm not saying, but it always has to be taken under medical supervision, or specifically if there's a certain disease condition, then it should not be taken just randomly. And what we found was that at that particular dose, the disease aggravated instead of curing. So that was a very good, interesting observation, a very significant one. Here we have used Brahmi Grita. So this is Brahmi Grita. However, when we feed these dry's mutants on Brahmi, so this is a dry's mutant, and these two over here are showing a fed at two different concentration of Brahmi Grita. And what you find is that compared to dry's mutant over here, there is rescue. So this is a very interesting part of work that is going on, where we have shown that Brahmikrita is actually able to rescue the Dry's mutant. And as I mentioned earlier, these Dry's mutant, the phenotype resembles polycystic kidney diseases. Okay, and so it is possible that if patients suffering from polycystic kidney disease (PKD), then they can it will be useful if we can test these for Brahmikrita for these patients. These are same, by different concentration of Brahmikrita, and you can see the level of uric acid is actually decreasing. So uric acid crystals, they can be visualized by polarized microscopy. You don't need any sophisticated instrument. It can be visualized very easily, and this is, you can see the reduction in the number of crystals upon feeding with Brahmikrita. So model for kidney disease. Okay, so one of the model for kidney disease is one of the ways by which kidney stones can be simulated in Drosophila. So there are certain diets which are responsible for making stone-like structures, and there are sodium oxalate. And so this part of the work is actually in collaboration with my student, actually Naveen. He's at SGPGI, so I have borrowed these. So we collaborate, and so we have almost we some papers are there where we both are there. And so these you can feed them on different agents, and this leads to the development of stone-like structures in Drosophila model, which represent the kidney stones, and they are calcium oxalate or sodium oxalate stones. And then you can try different drugs and see how it can be reduced. So this is crystal deposition by sodium oxalate. You can see these crystals, how they are deposited. This is control, and these are different concentration of crystal sodium oxalate, and you can see how it increases. Similarly, ethylene glycol, you can see the expression, the level of calcium oxalate crystals, they increase over here. These are all viewed by polarized microscopy. Okay, so I'll just skip this part. Okay. Now, another important research that you can do with Drosophila, we can induce type 2 diabetes. So, if you can feed these Drosophila larvae from the beginning on high sucrose diet, then you can have a condition which represents type 2 diabetes, and then you can study nephropathy, type 2 diabetes induced nephropathy. And the work plan is very simple. You just take male flies. Generally, we take male flies and we feed them on high sucrose diet. And then at different days, you can do different kind of experiment to see uh, the morphological changes of the malpighian tubules as well as the function. So function can be done by several different ways and morphological changes. So this is feeding of with 20 days with high sucrose diet. So you can see the morphology changes. Similarly, you have elevated glucose, trehalose, and glyceride levels, and this is insulin receptor. But I'll just skip this part. And as I mentioned earlier, that the organization can be very easily checked by looking at the actin organization, and you can see a difference. So actin organization is a way to understand whether there is any change in the malpighian tubules or not. Similarly, DLG, it gives you an idea about the arrangement and the polarity of the cells. So there's oxidative stress increased with high sucrose diet, and then there is increased expression of caspase over here. And then you have age, that is accumulation. This is advanced glycation end product. So this can also give you an idea about what level of damage is caused by feeding on sucrose. So since malpighian tubules are representing the kidneys over here, so we can understand how high sucrose diet affects the kidneys. So these are different ways by which one can, different methods by which one can see where exactly the damage has taken place. And a simple experiment to see the fluid secretion. So I don't know whether it's very clearly visible or not. One can use malpighian tubules and then at using different drugs and or different let's say high sucrose diet, and see whether the fluid secretion is affected or it is not affected. So this is, I think, this is all that I have to talk to you. And these are some of the students. They have left. Some are still here. And I would like to thank the fly community for sharing flies, different funding bodies, some previous students, and some students who did transcriptome bioinformatic analysis for me. Thank you so much.
Thank you, ma'am. Thank you for such an interesting and informative lecture from your part. And I hope that all of our participants have enjoyed a lot. So I now want to request our participants who want to ask any question in this particular topic, whatever ma'am presented. So please, anybody, raise your hands. Dr. Namrata Ray, yes, you can unmute your microphone.
Yes. Uh, Good morning, and thank you, ma'am, for a very lucid and simply explained lecture so that I, being a chemist, could also follow. Uh, but ma'am, can you please go back to that slide where you were talking about nanocarriers in AKI, and you talked about DNA nanostructures being one of the carriers. Ma'am, can you elaborate a bit more on that?
See, actually, DNA molecules, they can be, when you are, the polymerization can be done in such a way that you have, because, DNA, they are complementary regions. Okay. So the way the two strands, the complementation takes place, it helps to make a cage-like structure. Okay, so it is based on the complementation, the way you are, when you are synthesizing it, how one strand you synthesize and then you put the next strand. So that is, so it makes a cage. So this is a 4D DNA structure. Okay,
Right. Okay.
and then this cage, because DNA can enter into the DNA is it may not enter very easily, but being a nanomolecule, it can enter into the cells. So if you have any drugs loaded inside it, so the important thing over here is that these DNA molecules, they will not elicit any immune response.
Right.
Okay, so they are recognized, they are self, yeah.
Right. So, ma'am, when two strands of DNA, bind to each other through hydrogen bonding, so they create major and minor grooves. So the drug is loaded in those grooves, right? Then,
No,
and depending upon the structure of the drug, uh, the nano, I mean, the nucleotides are designed. That's what
No,
I
the
wanted to ask.
grooves drug may not exactly intercalate in the minor major grooves. The reason being because the size of the drug, it would be voluminous, or it may change the double stranded helical structure. Okay, so this
Okay.
change in helical structure may not be, it may be recognized by other repair enzymes, and that may be, that can uh, uh, lead to repair, and so it may break open this. So therefore, the drugs are actually inserted into the three-dimensional cage that is formed.
Cage.
Yeah.
Okay. Okay. Thank you, ma'am.
Thank you, ma'am. There is another question raised by Dr. Rajoshi Ghosh. He actually written in the chat box that what is the effect of dietary glucose on mitochondrial function and ATP production in empty cells?
In which cells? Empty cells.
Empty cells. Empty.
So, see, if Drosophila larvae, they feed on high sucrose diet, so they change the oxidative properties. So, the level of reactive oxygen species, the level increases. So, therefore, the mitochondria are affected. And in one experiment, what we observed was that the shape of the mitochondria has changed. They become more globular. If the larvae which is feeding on normal food, they have elongated shape, but when they feed on sucrose, the shape of the mitochondria changes. This we have not looked by TEM or SEM and TEM analysis, TEM, but by doing certain kind of staining, what we have observed is, when you stain, you have certain stain that antibody that we used was not staining, immunostaining. The antibody that we used was COP1. So this COP1, if you look at the shape of the mitochondria, they all appear globular, or you can see globular fluorescence compared to the elongated way which is it is seen in normal condition. So, of course, sucrose diet changes mitochondrial function. The exact mitochondrial function, the change that we have not looked into, but of course, yes, the ROS levels increases, which means that there are certain alterations in the mitochondrial function.
Okay, ma'am. Thank you so much. There is another question from Abhijit Bishwas. He asked that is carbon nanodots safe for human? It's a basic question, I think.
Yes, yes. Because many there are many of these medicines are now in the clinical trials, phase one and two. So they are safe, but of course, before using them on human, one has to check them at the lethal dose. One has to find out what is the dose, 50% lethal. And then for any drug, for any drug in that matter, you have to have certain trials, very stringent trials. And then if you don't see any lethality, because like rapamycin feeding, which we did with Drosophila larvae, so we tested them at different doses. So one dose in which it did not cause any developmental delay or fertility or lifespan in the wild type was used for mutants. For any even carbon dots, the way they are manufactured is, of course, it is not very harmful.
Thank you,
Thank
ma'am.
you, ma'am.
Okay, thank you. And now from Sujata Dhar Dotto, she asks that being a molting hormone, is there any role of ecdysone in insect immune response?
Yes, yes, yes. So I skipped that part. Yes, what we have shown is that ecdysone levels, because it changes during development. So, see, there are some reports, like in Bombyx Mori, where they say that ecdysone does not have any role or it has a negative role. But what we find is that the ecdysone levels, which is very high in the during pupal stage, at that time also malpighian tubules are produced. The antimicrobial peptides are produced, and we have also used certain ecdysone mutants to show that, and we have shown that ecdysone hormone is important for the expression of AMPs in the malpighian tubules. Yes.
Okay, ma'am. Thank you again.
We have
Now
also looked
we,
at some
yeah, sure.
downstream targets of ecdysone, like broad core, and we find that it is important for innate immune response.
Now we have another question from Annesha Chaudhuri. She asked that, ma'am, you told about actin deformation in Drosophila. Does the drug affect actin polymerization or deregulate depolymerization?
Drug. No, we have used phalloidin. No, what exactly you want to? No, I don't. I'm not getting the
Does the
question.
drug affect actin? Does the drug affect actin polymerization or deregulate depolymerization?
Which drug you're talking about?
You told about actin deformation in Drosophila. That's the
No,
first
no.
slide.
See, okay. What I'll just let me explain, and if it's still not clear, I'll again tell you. So what happens is, as I was telling you about malpighian tubule development. So initially, the malpighian tubules in the cells divide. So there's a division of cells. So you have a ball of cells, and as development proceeds, the same number of cells then they elongate to form the tubule. So this elongation of cells requires lot of cell rearrangement. Cells move from one place to another to rearrange align themselves in one tubular structure. So for this alignment, or let's say the realignment of cells, the actin organization is very important. Actin organization is very important for cell movement, morphology, and many things. So in malpighian tubules, actin is important for the elongation. So what we find is that if in rice mutant, the actin filaments are uh, very high, great expression of actin organization, which means, and therefore, what you see is rice mutants, they have the tubules are very cystic like. So that could be because of the disorganized actin. The drug that I was telling you, we have phalloidin. Phalloidin is a fluorescent molecule that goes and binds with the actin, which makes it visible. So binding of phalloidin to actin does not change the properties of actin.
Okay, ma'am. I think there's a last question from Manushi Mundol. She written that, Madam, have you got any positive outcome from the study of Drosophila, which can be applied to treat type 2 diabetes in human?
Yeah, so in Drosophila, the type 2, see what we have done is there's one compound called as pioglitazone. So normally this pioglitazone was used for some other disease. What we have done is we have studied the repurposing of drug. So when we have high diabetic condition, and if we feed pioglitazone to these larvae, we see the reduced level of lipid droplets. The obesity reduces, the size of lipid droplet reduces, and pioglitazone, along with in human, along with insulin, there's another peptide that's secreted that is called as IAP. So this IAP and insulin are co-secreted, and IAP is responsible. If there is an increase in the level of IAP, it causes tangles. That's amino peptide. It causes tangles and it causes Alzheimer's like disease condition. So what we have found is the pioglitazone, which is normally used for another purpose, if we use in this condition also with insulin, high sucrose diet fed larvae, there's a reduction of these tangles.
Okay, ma'am. Thank you so much. This is, I think, is all about from our question answer session. And ma'am, again, thank you so much for your such an interesting and informative lecture. And being a physics background from myself, I have also get enlightened from your talk about the nanoparticles to a bit
Okay.
extent. So
Thank you
again,
so
thank
much.
you all the participants for this session, and thank you, ma'am, again for your beautiful lecture. Thank you so
Thank you
much.
so much.
Thank you
Thank
all.
you.
So we are ending here right now. Thank you.
Thank you.
Thank you.
Our next, to the participants, you will please stay on this platform. Our next speaker is on the line now. We'll start right soon again.
So, good afternoon. I am here to introduce Professor Vishal Trivedi, who completed his PhD in structural biology at CDRI Lucknow, where he discovered a novel heme-containing peroxidase from the malaria parasite Plasmodium falciparum. He pursued postdoctoral research at Harvard University and Tufts University, Boston, USA, gaining extensive experience in intracellular signaling and immunology. Professor Trivedi has received several prestigious awards from Department of Atomic Energy, the National Academy of Sciences India, the Society of Biological Sciences, and the Association of Biotechnology and Pharmacy India. In the two lectures that will be delivered by him today, he will discuss about molecular cloning and different steps to clone an unknown protein. In the first lecture, we will cover the theory part, and in the second lecture, he will demonstrate the experimental details. So I will not take much time before between you and our respected speaker. So good afternoon, sir. I am handed over the mic to him.
Uh, Thank you, organizers. And uh, are you getting my voice right now?
Yes, sir. Yes, sir.
Okay. Yeah. So I'm very thankful to the Maulana Azad College for considering that I could be a valuable person and could be a resource person for giving a lecture. So as I could discuss with the principal, I think most of you are actually the young faculties, okay, in the department, right? Or I think the college, right? So the idea of this whole two lecture talk is that we are actually going to discuss about the molecular cloning, and I will try to discuss both aspects, the basic aspects of the theory aspects. And then we also going to discuss more about how you can be able to perform some of these experiments in your laboratory. The basic funda or the basic idea behind this whole exercise is that it should at least make you familiar. I'm not sure how many of you are from the biology background or how many of you are actually been exposed to the biology research. So it will just facilitate so that you should be able to utilize these techniques in case you require to achieve some of the objectives in your research career. uh, so I'm going to share my screen. So what I have done is I have divided the whole two lectures into in such a way that there will we can actually have a question hour sessions in between, and that may actually clear all your doubts. And even if you cannot get the answers or the questions right away, maybe you can ask me the questions when we start the second lecture also, post lunch. So I'm going to share my screen, and then we'll going to start. Uh, do you see a PowerPoint presentation?
Yes, sir.
Yes, it's visible.
Yes, sir.
Yes, sir. It's visible.
Okay. Okay. So what we are going to discuss during this first lecture is we are going to talk about the molecular cloning, and I'm sure majority of you probably could be fully aware of those who are from the biology background. So I have divided the whole thing into two parts. In the first part, we are going to discuss about the isolation of the gene of interest, right? And the second part, we are going to discuss about the post molecular cloning, how you can actually be able to uh, do the screening, how you can get the desired clone, and then how you can actually be able to use that clone for the protein production and all that, right? So what you see here is that this is the simple scheme of the molecular cloning, right? And in any of these schemes, what we have is we have the two counterparts, right? One is the counterpart where you are actually going to withdraw the gene of your interest, right? For example, the genome from where you want to have the gene of your interest. The second counterpart is where the carrier, the plasmid, right? And so imagine that we are trying to purify up or trying to get a gene from the from the human or mouse or whatever, right? So you have a genome. From there you have the multiple options. We will discuss all these options, how you are going to get the gene of your interest. Once you got this gene, right, and while you are doing this site-specific polymerase PCR with the site-specific primers, this gene is actually going to have the site-specific restriction sites. So when you cut this with the restriction enzyme, right, you are going to get the sticky ends on both the ends, right? The same procedure we have to follow even for the carriers. So I'm taking an example of the plasmids, which is for the bacteria, but it can be done even for the other carrier molecules or the vectors, such as mammalian vectors or yeast vectors and all that. And if you digest the plasmid also with the same set of restriction enzymes, it is also going to have the sticky ends. And then these sticky ends can be put together in a ligation reactions, and ultimately you are going to have the recombinant clone, right? So this recombinant clone you are going to generate, and then this has to be transformed into the bacteria, and then you are going to get the transformed bacteria. And this transformed bacteria is actually going to have the two different types of plasmids: the plasmid which is untransferred, like the plasmid where the gene of your interest is not present, or the plasmid which doesn't have the recombinant DNA. So then you are basically have to do the screening. So this is actually the transform. This is the actually the plasmid, which the bacteria which does not have the plasmid, and this is the the bacteria which actually has the gene of your interest. So ultimately, when you do the screening criteria, right, you're going to have the clone of your interest, and then you take these clones and do is all the downstream applications, right? These downstream applications could be related to the protein productions. It could be for the restriction enzyme, it could be for the structure and all that, right? Now if you see this scheme, right, you are actually have to follow the different types of steps in the molecular cloning, right? So this is the step number 1, this is the step number 2, right, and this is the step number 3, and this is the step number 4, right? And so in the step number 1, you are supposed to do the identification or the isolation of a particular gene, right? There are multiple approaches one can use for identifying a particular gene from the particular organisms, depending upon what kind of infrastructure is available, right, or what kind of information is available. Then once you identify the gene of interest, then you are going to go through with the detailed procedure of the cloning, right? So where you are actually going to do the gene amplifications, then you are going to do the the stationarization and the integration of your fragment into the vector, and then ultimately you're going to do the screening. Once you do the screening, you are going to get the clones of the particular bacteria where you are actually going to have the gene of your fragment. Once you have that, right, or once you have the clones, then you can actually take this DNA and put it into the delivery agent or into the host, right? And the host cells, depending upon the host cells, you can actually be able to do the transformations, like for example in the case of bacteria, or you can actually be able to do the transfection, for example in the case of mammalian cells. So this is for the bacteria, and this is for the mammalian cells. And we'll discuss all these steps in. And once you are done with the delivery, then again you are going to do another round of screening so that you can get the positive clones, and then these positive clones you can use for the overexpression, and then you are going to do the analysis of the overexpression. So we'll start first with the step number 1. The step number 1 is the identification and as well as the isolation of a gene fragment from the organisms. Now, when we talk about why we require this, right? So we require this because that you are actually going to encounter the different types of problems as far as the biological research is concerned, right? One of such problem is that you are actually going to have the two different types of cells. You are going to have the normal cells, you're going to have the cancer cells. Imagine that we have this kind of a hypothetical situation, that you have a normal cell and you have a cancer cell, right? Now, the normal cell and the cancer cells are varying in terms of their physiology, in terms of their cell biological applications, in terms of, so these two cell types are varying in terms of their morphology, right? You can see that there are so many morphological differences between the normal cell versus cancer cells. Then you can also have the cell signaling, which is also going to be different in between the normal cell versus cancer cells. And then you can also have the differences in terms of the DNA sequences. You know that the so many mutations are actually being responsible for the the production of the cancer cells from the normal cells and so on. Now, if we have a situation where we would like to identify a gene or I will say the set of genes which is responsible for the transformation of a normal cell into a cancer. Or if we want to identify the gene which is responsible for, I think I was listening the last talk, and I think it was about the actin remodeling or something, right? So suppose we would like to identify the cell signaling components which are responsible for the actin remodeling, right? Or uh, something like that. Okay. So if we want to do some unique component, we want to isolate a unique component from the cell signaling between this guy versus this, between the normal cell versus the cancer cells, we have to identify these genes or the gene product which is different between the normal cell versus the cancer cells. Now, if we have this kind of questions, right? When I say gene means I can also talk about protein, because every gene is responsible for the protein, right? So if we have these kind of hypothetical questions, it could be, this is just a simple one example. It could be many more examples. For example, if there is a twins, right? One is tall and one is short, right? Or one is tall, right? We can easily do the experiment and understand what are the proteins which are responsible for the length or the growth in an organism, right? So these kind of questions requires the isolation of that particular gene which is responsible for this particular type of phenotypes. Now, when we want to identify a gene, we have the multiple approaches which we can actually be able to use. So I have divided these approaches based on the whether a particular type of genomic sequence is known or not. So basically, it depends on whether the genomic sequences are known or whether the genomic sequences are not known, right? If the genomic sequences is not known, right, and remember that all these approaches are going to utilize the prior information, right? What are the information is available? For example, if you have the protein, right? If you are actually having the protein available, you know that these are the proteins there which are different between the two different conditions. Like for example, if you are doing the subtractive analysis, if you are doing the 2D gel electrophoresis, and you are doing the the control versus or the untreated versus the treated samples, and you know that okay, this is the protein which is different, right? But from the protein you cannot get the gene of your interest, right? So, or you actually have the antibody which is available, right? Or so those kind of, so these are the tools which are available, right, for you to say which approach you are going to use. So in the condition number one, when the gene sequence is not known, and this is very old story, right? And so when the gene sequence is not known, for example, if you are trying to identify the proteins or the gene from a organism for which the genomic sequence is not known, then you have the two multiple approaches within this also, right? The approach number one is where you are actually only having the information about a DNA fragment. For example, if you are having a DNA fragment which is different between the two conditions, or if you are having a very partial DNA fragment, then you can actually be able to utilize the genomic information what is available in that particular organism. So I'm not saying genomic sequences, I'm talking about the genome which you can isolate from that particular organisms, and then you can actually be able to prepare a genomic library. And once you are going to prepare the genomic library, you can actually be able to screen that genomic library utilizing this DNA fragment as the screening tool. And then ultimately it is actually going to give you the fragment, right, which is containing this particular DNA fragment. And that is, you're going to do the subcloning into a suitable vector, and that's how you can be able to identify the genes which are related to this particular fragment. Now, in the second situation, you basically have no information about the DNA fragment, but you have the information about the proteins, or you actually have the information about the antibodies. There are so many pathological conditions where people are not having the information about the protein, but they actually have the information about the antibody reactivity. So they see that the patient is developing, or when they take out the blood from the particular patient, right, they actually get the serum, and the serum actually contains the different types of antibodies. And these antibody composition is varying from the healthy individual versus the patients. And that could be one of the criteria. So you know that the antibody is going to recognize the protein, right? And ultimately you can actually be able to identify the proteins. So in that case, what you're going to do is you are going to prepare a transcriptome. So you're going to probe the transcriptome information, right? Which means you are actually going to probe the information about the messenger RNA, because we are talking about the proteins. And use this information to prepare first the cDNA library, and then you can use these antibodies to screen the cDNA library, and ultimately you are going to get a clone of your interest. And that clone you can be able to subsequently you can do the that cloning of that particular fragment into a suitable vector, and that's how you can be able to do the further screening. In the approach number 2, when the genome sequence is known, right? For example, for most of the organisms, we are actually doing the genome sequences, right? Then you actually have the genome sequence. So once you have the genome sequence, what you can do is you can actually be able to prepare site-specific primers, right? And once you have the site-specific primers, you can use those site-specific primers to perform the polymerase chain reactions, and the polymerase chain reaction is going to give you the fragments, and these fragments are actually going to have the sticky ends on both the ends, and then you can actually use these sticky ends or the restriction sites to clone this into a suitable vector. So these are the three approaches, or these are the three important tools what are available for you to isolate a particular gene of interest, depending upon what could be your starting material. If your starting material is a DNA fragment, you can actually be able to use the approach number 1, where you are going to use the genomic library. If the starting material is a protein, or if the starting material is an antibody, then you can be able to use the cDNA library, and you can actually be able to use the transcriptome informations. If you have the genome sequence known, then the life is very easy. You can actually be able to design the site-specific primers, and that actually will give you that particular fragment from the genome, and that you can actually be able to put it into the expression vectors, and that's how you can be able to produce the protein, right? I hope everybody is clear about this, right? So if you are not very clear, you can actually be able to note down your doubts, and then we will discuss about your doubts in subsequent. So let's start first discussing about the approach number 1, right? So in the approach number 1, the first information is the genomic library, right? How you can be able to prepare the genomic library. So this is the construction of the genomic library. So it actually has the multiple steps, right? In the step number 1, you are going to do the isolation of the genomic DNA. Then once you are going to have the isolation of genomic DNA, then you can actually do the restriction digestion of the genomic DNA, so that you can be able to generate the different types of fragments. Then these fragments you can be able to do the purifications, right? And then once you are going to purify these fragments, then you can actually be able to do the ligation into a suitable carrying vectors. So in this case, we are taking the YAC vector, or the yeast artificial chromosomes vector, and then it is actually going to transform into the yeast, and then that's how you are actually going to get the genomic library, right? So basically what we are doing is we are taking the human DNA or the genomic DNA, then we are digesting this with any of the common restriction enzymes, such as in this case, we are taking an example of EcoR1. So what the EcoR1 is going to do is it is actually going to take the DNA, and it is actually going to chop the DNA into the small fragments. Now, these fragments are actually going to have the EcoR1 site on both the ends, right? Because only we are doing the single digestion with the EcoR1, right? So it is actually going to have the EcoR1 sites on both the ends, right? Now what we are going to do is, same way, we are going to take the YAC vectors, or the artificial chromosome vectors. We'll digest that also with the EcoR1, right? And the EcoR1 is going to give you the fragments. Now, when you combine them, right, these fragments where you have actually the CoR1 sites will ligate into this particular YAC vectors, and then you are going to do the transformation into the yeast, and ultimately you are going to get the colonies. So you will actually get the colonies of the yeast, and each colony is actually going to contain the different types of clones, right, which actually contains the different types of fragment. And that is what the purpose of preparing a genomic library, right? If you want to search a particular DNA sequence in a particular genome, it is very difficult. So what you are doing is you are taking the genomic DNA and then you are splitting that into a small fragments, and then you take these individual fragments into the individual colonies. So basically what you have is you have the individual colony which represent this particular fragment, you have another colony which represent to this particular colony, and so on. So that is what is going to do. So when you are going to do a screening into this particular colony, right, these colonies, right, and suppose this particular colony is going to give you the indication that okay, this is the clone, right, that means you basically can extract out this particular colony and you can actually be able to get the idea, right? Because all these clones are representing the different site within the particular DNA, right? So the step number one is actually the isolation of the genomic DNA, right? So isolation of genomic DNA is having the 5 or 6 steps. So in the step number one, you are going to do the lysis of the cell with the help of the lysis buffer, or which actually contains the detergent, right? Remember that in the step number one, you are actually going to use either the cultured cells. You can actually be able to use the tissue, right? If you use the tissue, for example, if I want to understand what happens to the liver when it is getting the infection of the hepatitis, or what genes are responsible, or what are the genes are responsible for the liver, when it actually, getting the infection of the hepatitis virus, then what we are going to do is we are going to get the liver, right, from a particular patient, right, or a fragment of liver, right? In that case, what we are going to do is first we are going to convert the liver tissue, right, into the cell, the individual cells, right? And then you are going to perform the step number one, that is the lysis of the step, right? So you are going to lyse the cells with the help of a lysis buffer, which actually contains the detergent. And you know all that how the detergent is going to lyse the cells, right? It is actually going to dissolve the plasma membrane, or it is going to dissolve the lipids, right? And once the lipids are dissolved, they are actually going to make the pores, right? And that's how these cells are going to be lysed. Once the cells are lysed, it is actually going to release its content, right? So if it is a eukaryotic cell, it is actually going to release all the material, right? So it is going to release the protein part, it is going to release the DNA, what is present inside the nucleus, and it is also going to release the lipids, carbohydrate, and everything. So then basically what you're going to do is, and you know that the eukaryotic DNA is also going to couple with the protein, right? So you are going to incubate the cells with a digestion buffer containing the proteinase K, SDS, to release the genomic DNA from the DNA protein complexes. After this, you are basically going to have a mixture of protein, DNA, lipids, and partially you are also going to have the carbohydrates. Then what you're going to do is you're going to precipitate the genomic DNA by the absolute alcohol, right? And then once you got the DNA, right, this DNA is actually not going to be pure, right, because it also has the contamination of the proteins, it also has the contamination of the lipids. Then you are going to do the purification, right? So purification you are going to do with the help of the phenol chloroform mixture, right? And the chloroform mixture has two phases. One is the aqueous phase and the other one is the organic phase. So in the aqueous phase, you are going to have the your DNA, and whereas in the organic phase, you are going to have the proteins, and that is actually going to remove the protein from the your DNA. right? Now your DNA is free, and then you are going to precipitate the DNA, and again with the help of the absolute alcohol, and then you are going to analyze this DNA onto agarose gel so that you can be able to see whether the your genomic DNA preparation is good or not. So how you will know that the genomic DNA preparation is good or not, right? First is genomic DNA is very heavy, right? So it is heavy in terms of, because it has a lot of, it has a mega base pairs, right? So all genomic DNA of all the organisms are in the mega base pair range, right? So they are very large, right? So they are actually going to run very close to the wells, right? So they will run very close to the well, right? The second is the genomic DNA, which is intact and which is not having any kind of degradations, should be run as an intact band, right? So, for example, in this case, right, it's running as an intact band. It should not have a smear. It's not like the genomic DNA is running like a smear. If you are running the genomic DNA, which actually has the the fragment like this, that means it actually has the the DNA fragments, right? And that is not good for preparing the genomic DNA, because whatever the fragments are present here, right, they are actually going to be coming out from the genomic DNA, and they may not good because of the simple reason that this kind of information is going to be lost. For example, in this case also you'll see that there is a very partial or there is a small level of degradation into the genomic DNA, but that is still be okay, because that happens because of the because of the movement and all that, right? So once you are going to isolate the genomic DNA, then the next step would be that you are going to do restriction digestion. So so isolation of genomic DNA is a multiple step process, right? So in the step one, you're going to do the lysis. Then in the step two, you are going to do the digestion, right? And subsequently, you are going to do these isolation procedures. Once you are going to have the genomic DNA, then you are actually going to, so genomic DNA, then you are supposed to generate the fragments, right? How you're going to generate the fragments? There are multiple ways in which you can be able to generate the fragments. The first is you can actually be able to use the restriction enzymes, right? So you can actually be used, the genomic DNA can be digested with a frequent DNA cutting enzyme such as EcoR1 or BAMH1 or SAW3A to generate the random sizes of the DNA fragment. The criteria to choose the one enzyme or the pair of enzyme is depends upon whether you are getting a reasonable size DNA fragment or not, right? So so how you are going to say whether the reasonable size is you are getting the reasonable size or not? So you can actually be able to use this particular equation. So what this equation is saying is the number of clones, right? Remember that number of clones is actually going to increase your probability of getting your fragment individually. So for example, if I have a genomic DNA like this, right, and imagine that we have the multiple genes like this, right, what our objective would be that, so suppose this is the gene number 1, this is the gene number 2, this is the gene number 3. Now our objective is that the gene number 1 should get cloned into singly, right? And gene number 2 also should get singly, right? And gene number 3 also should get singly. So that when you are going to isolate, right? Suppose this is for the gene number 1, this is for the gene number 2, and this is for the gene number 3, right? So if I get this fragment, right, or if I get the positive signal from this particular colony, then I will be sure that I am, this is for the gene number 1, right? Imagine a situation that with the same genome, I am generating the fragment, but the fragments are of this size, right? A fragment of is this size, which actually contains the gene number 1 and 2 and 3. And in that case, what happens is it is actually going to generate the confusion, because then even then you will not be able to be sure whether my probe is interacting with the gene number 1 or gene number 2 or gene number 3. So the smaller the fragments, not the smaller than your gene of interest, right? But it should be good enough, right? So the ideal fragment is that you are going to have the gene number 1, and then you can have the small portion of gene number 2 and small portion of gene number 3, right? So if that is the case, then that should be the ideal case, right? So so idea is that you generate as many fragments possible, so that the number of fragments you increase, the number of clones you are going to get. And then as you increase the number of clones, it is actually going to have the same probability of that you will get your gene of interest. So apart from these restriction digestions, you can also use the genomic library. You can actually be able to generate the fragments using the mechanical sharing. For example, you can actually be able to do the vertexing, or you can actually be able to do the sonications, right? So if you do the Bass sonications and you can do the vertexing, that will generate the mechanical sharing, and that also is going to give you the product, the fragments. The only the problem is with the mechanical sharing, the site specific, the sticky ends would not be present. So the cloning these fragments into a vector would be a problem. Now we have taken an example. For example, if there is a genome, if there is an organism of genome size like 2 to power 10 to power 7 kb, then and if you are having the individual size which is like 20 kb, then the number of fragment would be 10 to power 6. So you can just simply divide this, you will get this number, right? So in reality, this is the minimum number of fragment which is going to represent the entire library, whereas the actual number remained has to be even larger than this. And that's why the probability of finding a particular sequence is this, right? So you can use this formula, and you can actually be able to know whether the genomic library what is have prepared is good or bad. Then the step number 3, you are going to clone these fragments into a suitable vector. So we have multiple types of organisms. So you see that we have smallest organism like the mycoplasma, and then we also have the bigger organisms like the flowering plants, right? And the flowering plants are actually having a genomic size in the range of 10 to power 11 base pair, whereas the mycoplasma has the genomic DNA in the range of the 10 to power 6. So you see that there's a huge diversity. So you're basically going to have the different types of fragments, and then you can actually be able to generate these fragments. You can actually be able to put it into the suitable vector. What vector you will use for preparing the genomic library depends upon the size of the genomic DNA of that particular organism. So because every vector is actually having a unique size of carrying size, right? For example, if you have a plasmid, plasmid cannot carry more than 15 kb, 15 MB actually. Similarly, you can have the Phage lambda, which is going to carry the 25. Then you have cosmids, bacteriophage, you have the BAC, and then you also have the YAC. So you see the YAC and BACs are actually going to have a very large carrying capacity. So if you are trying to generate the genomic library from the plants, or if you are trying to generate the genomic library from the different types of bigger organisms like the humans and dog and all those, then you have to use the BAC or the YAC as the carrier, right? Whereas it's for the smaller organism, for example, if you are trying to prepare the genomic library from the MTB, then you can use the plasmid also, or you can use the YAC also. So the after the post transformation, you are actually going to prepare the genomic library, right? And I think I have explained it in detail that genomic library means it is actually going to be the fragmentation of the genomic DNA and the cloning into the individual clones, so that it is actually a representation of the genome. It is actually going to be a representation into the multiple clones, so that you can actually be able to isolate the gene of your interest from that particular clone. So that isolation part we will discuss in a later on. Now let's move on to the next step. And the next step is you can actually be able to do the construction of the cDNA library. So in the construction of the cDNA library also has a multiple steps. For example, so remember that the cDNA library, we are actually trying to exploit the transcriptome information. So from the genomic library, we are actually utilizing the genomic informations, whereas in the cDNA library, we are using the transcriptomic information. So first is you can actually be able to isolate and collect the messenger RNA. The second is from the messenger RNA, you are going to prepare the cDNA, and then the cDNA is going to be inserted into the vector of your interest, and then you are going to get the colony, and then these colonies you can actually be able to use it for the different types of screening and isolations. How you are going to purify the messenger RNA or the messenger RNA pool of particular from the particular cell, for example the normal cell versus the cancer cells. So if you see the structure of the messenger RNA, I'm sure everybody know about the structure of the messenger RNA. So messenger RNA has the the very distinct peak, right? You have the 5' UTR, and above before the 5' UTR, you are actually going to have a cap, and then next to that you are going to have the coding sequence. So this coding sequence is going to be unique for each protein, right? And then you have the 3' UTRs, and after that you are actually going to have the poly A tail, right? Now this poly A tail is a very important tool if you want to purify the messenger RNA of a particular cell, right, at a particular conditions, right? So for example, if we have the cancer cells, right, and we have the normal cell, right, and we have the cancer cells, right? Then what we can do is we can take the normal as well as the cancer cells, right? We lyse them, and then we can actually utilize this information like the poly A tail. So poly A tail is not present in any other the messenger RNA sequences, right? It's only present onto the messenger RNA. So you can use this as a criteria to isolate all the messenger RNA from the particular cell. So what you're going to do is you can take the cell, right? You can take the cell or you can take the tissue, right? Then you put it for the lysis, and once you're done with the lysis, you are going to get the lysate, right? And from the lysate, what you're going to do is you're going to put it into the binding buffer, right? And then you add the oligo DT beads, right? So what oligo DT beads are, right? These oligo DT beads are the beads, right? So you have the silica beads, then it has the particular DNA, and that DNA is actually containing the T nucleotides, right? So it actually has the thymine, right? And remember that the A is having the very specific interaction with the T, right? So when you are putting this into this and then you are mixing them, right? Then what happen is that the poly A tail, like the poly A tail, what is present onto the messenger RNA, right? It is actually going to interact with this poly T tail, right? So what will happen is that all the poly A which is present on the messenger RNA is going to bind, and then it is actually going to interact with this, right? So in the first step, when you are doing the binding experiments, right, poly or with the oligo DT beads, right, this will happen, right? So these beads are actually going to bind all the messenger RNA, right, from the lysate. And then you are going to do the washing. So you actually can do the washing with the with the high salt buffer, right? And so that is actually going to remove the non-specific interactions. So but since this is very specific interactions, it will not be sensitive for the high salt buffer. And then you are going to do the elutions, right? So you can actually be able to do the elution with the help of by changing the pH and all that, right? And then ultimately you can be able to collect all the messenger RNA, right? And once you have the pure messenger RNA, right, from the particular cell, right, you can use this for preparing the cDNA library. So you can actually be able to use these messenger RNA for preparing the cDNA, and then from the cDNA you can actually be able to prepare the cDNA library. So in the step number 2, you are supposed to prepare a complementary DNA, right? So in the complementary DNA, what you are going to do is first you are going to do a synthesis with the reverse transcriptase, right? then you are going to have the removal of the RNA templates, and then you are going to have the second strand synthesis, which means basically what you have is you have this, right? You have the messenger RNA with the poly A tail, right? And then the step one, you are going to prepare the the DNA, right? So utilizing this, you are going to prepare the DNA, right? With the help of the reverse transcriptase, right? And then in the second step, you are going to do something so that the RNA is going to be removed, and then you are going to do the another synthesis so that you are going to have the double stranded DNA, and that double stranded DNA is going to be cloned into a particular vector, and that's how your the cDNA library is going to be prepared. So there are multiple methods of preparing the cDNA library or the cDNA of a particular messenger RNA. We are not discussing about all those methods, right? So this one of the popular method is called as the homopolymer tailing. So in the homopolymer tailing, this is the messenger RNA what you have, right? This is the messenger RNA where you have the A, right? And so what you're going to do is you're going to put the oligo-DT primers, right? So what will happen is the oligo-DT primers are going to bind here, right? So just like this, right? And then you are going to put the reverse transcriptase and all the nucleotides, right? And then what will happen, the reverse transcriptase is going to take this information and it's going to synthesize the cDNA. Once you have, so you basically what you have is you have the RNA on this side and you have the DNA on this side, right? Remember that the RNA is having the ribose sugar, the DNA is having the deoxyribose sugar. There's no other difference, right? And then what you're going to do is you're just going to put this whole into alkaline conditions like the alkaline sucrose gradient. And once you're doing with this, right, alkaline sucrose gradient, the alkaline condition is actually going to destroy the RNA, right? Because of the simple reason that RNA is actually having the ribose sugar, right? So I'm sure majority of you probably know about the RNA, right? RNA is having a ribose sugar, right? This is the ribose sugar. And so what you have here is you are actually having the 2' OH, which is completely absent in the case of the deoxyribose sugar. So this 2' OH is not present, right? The DNA is only having the 5' OH and the 3' OH, right? So this OH is responsible for the cyclization of this particular RNA molecules, and because of that, the RNA is very susceptible for the alkaline lysis. So when you put this whole DNA protein hybrid, DNA RNA hybrid into the alkaline conditions, it is actually going to hydrolyze, and it is only going to hydrolyze the RNA part. It is not going to hydrolyze the DNA part. And then once you are done with that, then you are actually going to have the cDNA. Then you put the cDNA into the another second strand synthesis, and it is actually going to give you the duplex of the cDNA, which means it is actually going to give you the double stranded DNA. Now this double stranded DNA can be inserted into the vector by the linkers or the bihomopolar tailing. And then second method is the Guwahati method, which I probably will not going to discuss. Okay. So far what we have discussed the approach number 1, right, where you can actually be able to utilize the genomic DNA. So you can actually be able to prepare the genomic library. In the approach number 2, you can actually be able to prepare the cDNA library. And once the library is prepared, then you can use the different types of screening criteria to screen the clone, which actually contains the gene of your interest, right? So how you're going to screen? So I said, right, if you have the DNA fragment, so you basically have 3 tools. One is you basically have the DNA fragments, which is present and which is different between the 2 different types of conditions, or you can actually have the proteins, or you can actually have the antibody, right? So what you are going to do is you're going to put this whole library into the vector by the linkers or the bihomopolar tailing. Okay. And then second method is the Guwahati method, which I probably will not going to discuss. Okay. So far what we have discussed Okay, or number 4, you can actually have some kind of activity which is present in one condition and which is completely absent in the another condition. So these are the 4 tools which are available for you to screen, right? So how you're going to screen the genomic library or the cDNA library? Remember that the genomic library is actually having the genes which are expression genes or the non-expression genes, right? So it is actually a representation of the whole genome. So it is actually going to be a representation of the expressive genes and the non-expressive genes, right? So there are actually fragments which does not express and which does not produce a protein. Whereas the cDNA library is only going to be a representation of the expression genes, right? So it does not represent any of those non-expression proteins or non-expression genes, right? So that is something what you have to keep it in mind, okay? So how you're going to do the screening, right? So first is you can actually be able to use a DNA sequence, right? Or you can actually, so this protein can be used to search both genomic library as the cDNA library to identify a particular gene, right? And in the second approach, you can use a particular protein with the immunogenic epitope. So if you suppose got some antibodies, so that can be used also. But that only can be used for screening the cDNA clones, because now majority of the genomic library is actually having the either they will have the non-expressive fragments, or they may have the full. They may not have the full length gene actually. The third is the enzymatic activity. So this property, you can actually be able to exploit only for screening of the cDNA library, not for the genomic library. So for the genomic library, you can actually be able to use only the DNA sequence, or to some extent you can use the antibodies. How the DNA hybridization can be used as a criteria. So basically what you have is, you know that the A is making the complementary sequences with the T, and G is making the complementary sequences C. So they are very specific, right? And we are having a very genuine reason why the A is making a pair with T and G is making a pair with C, right? So in the genomic library, it's screening by the DNA hybridization. What you are going to do is, the first is this is your genomic library. Okay, so this is your genomic library. Okay, so first thing what you are going to do is, you are going to make a replica plate. Okay, so this is your master plate from where you actually have the all sort of colonies, right? For the simplicity purpose, I'm just showing very small number of colonies, but that may not be the case. When you are going to deal with the actual plate, you are actually going to have the thousands of colonies, right? So in the step number 1, what you're going to do is, you're going to make a replica plate, so that your master plate or the genomic library should not get destroyed. Okay, so what you're going to do is, you're going to take the small nitrocellulose membrane, put it onto your plate, and then you transfer these patterns, right, into a replica plate. So you basically going to do all the operations onto a replica plate. With replica plate, where you are going to have a similar kind of patterns, but they are going to be a representation of the master plate. Okay, then the step number 1, you are going to do the lysis of the cell, and once you lysis the cells, what will happen? The cells are still be there, right? And you actually going to hybridize them, right, with the DNA, right? So basically the cells will still be there, and they will contain your genomic DNA, right? But or the fragments, right? But they will not going to be come out. Okay, and then what you're going to do is, you're going to add the radioactive probes, and then you do the hybridizations. So this particular DNA, the radioactive probe, will go and bind to a particular colony. So it's when it binds to this particular colony, right, it's actually going to give you the signal into the autoradiograms. And that's how, now suppose you got this right, then you can actually be able to get this colony from the master plate, right? So you can actually be able to extract out the colony from the master plate, and then you can do the subculturing to see whether this particular colony is pure or it is still having the multiple clones, because it's possible that a particular DNA could actually, multiple DNA could be present in a particular bacteria, or this colony may contain a fragment which is containing not the single gene, it may have the multiple genes, right? Or it may actually have a fragment of the neighboring genes, and your protein should be interacting with this one, not to this one. So this is actually intact, but it also has the neighboring fragments, and these neighboring fragments are actually giving you the signal. So for this whole screening of hybridizations, you basically require the radioactive probes. And you radioactive probe, how you're going to prepare the radioactive probe? So what you're going to do is you just take the DNA of your choice, right? And then you do the random primer methods, right? So you can actually be able to add the oligonucleotide primers, right? And you do the hybridizations, you do denaturations, and then when you are going to do the DNA synthesis with the clonal fragments, and it is actually going to incorporate the radioactivity into these small fragments, and then when you do denature, you are going to get the radioactive fragments, right? And these radioactive fragments, then subsequently you can do the gel filtration, and you can be able to purify these fragments, and you can use them for screening the genomic library. So this is the the different types of steps. So first the DNA is going to be denatured, generate the single stranded DNA templates, and then the random primer is added and allowed to anneal to the template, and it will anneal to the random positions to the sequence at the multiple places, and the primer will anneal to the template strand, and now Clino will start the synthesis of new DNA strands. And once you have this new DNA strand is going to be synthesized, then you actually can add the radioactive nucleotides, and the newly synthesized DNA will have the short stretches of multiple labeled DNA probes. The second method is the terminal transferase method, and that also we can use. And then, so first you are going to prepare the replica plates, then you blot these, and then you do the hybridizations, and then you do the development of the blot. So once you do the development of blot, it is actually going to give you the position of that particular clone, and then you can actually be able to extract the colony from the master plate. The second method, you are going to do the screening by the immunological methods, right? So in the screening of the immunological method, you are going to, the first step is same, right? From the master plate, you are going to prepare the replica plate, and then you are going to lyse the cells, and you allow the protein to bind. So basically when you lyse the cells and you ensure that the protein will still stick to the same position where the colony was present, then you are going to add the primary antibodies, and then you are going to do the washing, and then you add the secondary antibodies, and then what will happen, the primary antibody will go and bind to this, right? And the secondary antibody will go and bind to the primary antibodies, and then you do the the in between you are going to do a washing and all that, and then ultimately you're going to develop this with the help of the the colored substrate of the HRP, and it is actually going to give you a signal here. So this means this is the colony what is very responsible for this particular type of phenotype. Then what you're going to do is you go to the original plate, and then you can actually be able to extract this particular colony, and you can actually be able to extract the particular DNA fragments, and that's how you can actually do the cloning and subcloning into the suitable vectors. The similar kind of procedure you also have to follow by the screening by the enzymatic method. So this method is based on the ability of protein to exhibit a particular enzyme activity. This method is not very specific, but it allows to identify a class of protein with the known enzymatic activity. Now, once you have identified the clone, right, from the clone, how you are going to isolate the gene, right? So for that, you are actually going to, so once the position of a clone is known, it is extracted from the master plate and the plasmid is isolated. In few cases, the clone is further diluted to check the homogeneity of the clone. The purity of the clone and the presence of clone is further tested with the PCR using the sequence-specific primers. So what we have discussed so far, we have discussed about the approaches when the genomic sequences are not known, right? So we have discussed about the genomic library, we have discussed about the cDNA library, okay? And we discuss about how you can be able to prepare the genomic library, how you can be able to prepare the cDNA library, and once you prepare these libraries, you can actually be able to do the screening with the help of the different types of tools. So you can actually be able to use the radioactive DNAs, you can actually be able to use the antibodies, you can actually be able to use the enzymatic activity assay to screen. And once the screening is done, then you are going to get the suitable clone, which actually contains the fragment of your choice. And from this, you can actually be able to extract the DNA of your choice, and then you can actually be able to do the subsequent cloning into the vector, and then you can use this for the downstream applications. So we are almost halfway down, right? In case you have any queries, or you can ask me one or two questions, then we can actually be able to move on.
Yeah, hello sir. This is Prajit Nambiar from Mumbai. I just wanted to ask you, like, how do we lyse the bacterial cells on the plate for DNA hybridization without disturbing the colonies?
Um, okay, so when you are actually going to use the colony, right? The You are actually going to because everything you are going to transfer it onto the the replica plate, right? So the first step itself is that you are actually going to fix them in such a way so that the proteins or the content is not going to be released. So first step is you are going to do the fixing, right? And once you fix them, right, you can actually be able to do the lysis, okay? And that lysis will not, it only going to make the pores into the membrane, but it will not allow the protein to come out, protein or DNA.
Okay, okay. But how do we fix? Like, do we fix just the DNA on the membrane?
No, no, no. You fix the whole cell. You'll dip the membrane into fixation, or in some cases, what people do is they actually treat it with such a diluted condition of the detergent that it only makes the pores good enough for the liquid to come out, or the proteins to interact with the your probe, but it does not allow these biomolecules or the bigger molecules to come out. See, when you have so for example, like for example, this you have a cell, right? This right. If I treat this with detergent, I have two options. I'll get the two outcomes. One, where the complete plasma membrane is going to be removed, okay, and it is actually going to release the content. The second is, in one case, in other cases, if I do the controlled detergent treatment, then it is actually going to make the the gap like this. So it is actually going to do the the spores, which will allow the probe to go right, which will not, but it will not allow these protein molecules to come out because these pores are going to be very small.
Okay.
Is it clear?
So one more thing, like we normally do the RPT using the replicating block, which is in a very old method. So when you're using this nitrocellulose membrane, are there any specific grade or specific marking on them to know, like grid-like pattern or something?
Can you repeat one more time?
Like does this nitrocellulose membrane, like does it come with any specific grid-like structure or pattern so that we can identify which colony we are marking?
Okay, okay. Yeah, no, no. When you are going to have the master plate, right? Master plate itself will have the, the dots like this. You can actually be able to prepare the dots like this.
Yeah.
So suppose this is your master plate, right? So what I'll do is I'll just put a paper sheet like this and soak it, right? So if I put the master plate like this, right? the moment I will peel it off, right, I can put the position of these three molecules. These are not colonies actually. These are only the dots which are there on the master plate. I will just put the similar place. I will put a dot in my paper.
Okay. Thank you so much.
Anybody else? You can have 5 minutes. Question, then we can move on, because I don't want to keep going and then ultimately people will have a problem of following, right? Shall we continue?
Yes, yes. If anyone has any question, please put or raise your hand. I think.
Okay. I think I will.
Okay.
I'll
No
continue.
one.
Yeah.
Okay. Okay, sir.
Yeah. Okay. So next we'll talk about the third thing, right? When the genomic sequence is known, right, then the life is very easy, right? You have the genomic sequence. You can actually be able to prepare the site-specific primers, right? And you can actually be able to do the PCR, right? And that's how you are going to get the amplified product. And all these fragments are actually going to have the restriction site on both the ends. And when you digest them and that's how when you digest them with a particular restriction enzyme, you are actually going to have the sticky ends, right? You're going to have the sticky ends, right? And these sticky ends can be used for then to clone into a particular vector. For the PCR, right? So that's why it is very popular, because it is easy, right? Remember that the preparation of genomic DLA and preparation of cDNA library is a very difficult and a tedious process, with the only reason that you're supposed to have the desired number of clones, right? So when you do the restriction digestion, restriction digestion of the genomic DNA, sometime it will give you a very large fragment, and that subsequently is going to reduce the number of clones, and then you have to discard this, because the probability of finding your fragment will be very low, right? In other case, if the fragments are too small, then you are actually going to get the large number of colonies, and then it is actually going to have another problem of that multiple clones are actually going to light up, right? Remember that only few clones should light up when you're doing the genomic library screening, right? If the 1000 clones are going to light up, then it is very difficult to isolate those 1000 clones and filter out which one is the real one and which one is the fake one. And the other problem with these kind of approaches is that they are non-specific, because you are using the tool which is very blindly, you're using antibody, you are using the DNA and all that, so it is not that specific. So, and because of that only, the people thought of doing the genome sequencing techniques and the utilization of PCR, right? So what is PCR? PCR is a technique which actually allow us to amplify DNA, double stranded DNA fragments with the same identical size and the sequence. So remember that you are supposed to have the identical size and the identical sequence, and you are actually going to use the enzymatic method of the cyclic condition. So for example, if this is your template, right, what the PCR is going to do is, so you actually have only one copy of template. It is actually going to allow you to generate the multiple copies of the same fragments, right? So that you can actually be able to have the more different type, more amount of DNA, so that you can be able to use this for the different types of applications. And while you are doing this, for the cloning purpose, people are putting the restriction sites on both the ends, so that it becomes sticky also, and that's how you can actually be able to use these additional tools for cloning into a particular vector. So I'll just very quickly go through with the basic idea about the DNA. So DNA is composed of the two complementary nucleotide building blocks chains. You know that the DNA is having the 5' nucleus and 3' and the 3' to 5'. So it is a double stranded molecules, and the nucleotides are made up of the phosphate and the sugar and a nitrogen base. And talking about the nitrogen bases, so you have the purine bases, you have the pyrimidine bases, and all these four bases are repeatedly treated by hydrogen bonding between the nitrogen bases, and the linking of the two complementary strand is called as the hybridization. So DNA is complementary in nature, and that actually makes the DNA as a very, very unique molecule. So what is mean by the complementary is that you are actually going to have the in terms of base pairing. So A is always going to make the 2 hydrogen bonding with T, and G is always going to make the 3 hydrogen bonding with the C, right? So for example, in this case, right, this is the primary strand, right? So this is the original strand. So wherever you have the G, because the G is making a pair with C, it is actually going to make a pair with C. Wherever you have the C, it is going to have G. So for example, this is G, you have C, G, C, C, G, like that. So this is being called as a primary strand, and this is going to be a complementary strand. And this actually allows not only the synthesis, but it also allows the propagation of the information from the primary strand to the complementary strand. So you can imagine that this primary strand could be belonging to your parents, right? And this is a complementary strand which is belonging to the kids. So that is the reason that the genomic DNA is being chosen as the as the molecule for carrying the genetic informations, because of this complementary in nature. Now what we have is we have this information available, and we want to generate this information, right? So we basically have the DNA information available, right? And we also need a machinery to just generate the complementary strand, and that's how you can actually be able to achieve the the PCR amplifications. Now, if you see that this process is not new, right? This is process the cell is always doing, right? It is taking the information, the primary information from the template strand, and it is generating the complementary strand, right? With the help of the different types of enzymes. So, what are different types of enzyme? It is actually using the sorry about this. So I will just skip this because this is not very important, right? So okay, so what you see here is that inside the cell you basically have the three different types of steps. You have the initiation, you have the elongations, and then you also have the terminations. So in the initiations, the protein is going to bind the DNA. They will open the double stranded DNA. Remember that we have the double stranded DNA, right? So then the step one, you are basically going to have the single template, right? Single template, so that you can be able to generate the complementary strand. Then in the elongation phase, the proteins are going to bind to this particular single stranded DNA, and then they are actually going to start synthesizing the nucleotides based on the information of the nucleotide what is available onto this. So if you have G, it is going to put C. If you have A, it is going to put the T, and so on. So that's how it is actually going to do the DNA synthesis. And once it reaches to the corner or it reaches to the end of the DNA, it is going to terminate by the stopping of the DNA synthesis, and the protein, whatever is the machinery is being present on this particular single stranded DNA, is going to be released from the DNA, right? Now, if you see that this process, so basically you have the different types of proteins. You have the helicases, you have the primase, you have the SSB proteins, you have a DNA polymerase, and then you also have the T3 proteins. So the first is in the elongation phase or in the initiation phase, the helicase will come and it will separate out the DNA, right? So it is actually going to separate out the DNA and it's going to generate the single stranded DNA. Then the primase will come and it will synthesize the primer, right? So it is actually going to prepare the primers. because of the simple reason that the DNA polymerase cannot sit onto the single stranded DNA and synthesize the de novo synthesis. It actually requires the double stranded template. So single DNA polymerase has to sit onto a double stranded DNA, and then only it actually can synthesize the DNA. So and then primase will come, it will synthesize the the primer, right? And then the SSB will come and sit in between these two strands, right? So that the SSB, so whatever the strand you have made the single stranded should not re-anneal, right? And then the DNA polymerase will come onto this DNA hybrid, and it will synthesize the new strands. And while all this going on, the T3 proteins are also present, and that will also going to stabilize the polymerase reactions. So what you see is that the DNA synthesis is very simple. You have to do the initiations, you have to do elongations, you have to do terminations. But that is not very easy when you are talking about the in vitro system, right? Because in the in vitro system, uh, the the balancing between the different types of proteins. So remember that we have the helicases, we have the primase, we have the SSB proteins, we have DNA polymerase, and then T3 proteins. But what we don't know is, or what we cannot control inside a test tube is that when the helicase will come, when the primase will come, when the SSB will come, when the DNA. Because if you take a append off, right, and if you add the helicase, if you add the primase, if you have SSB, if you have everything, right, then they will not work in coordination with each other, right? In a cell, you basically have the machinery, you have the regulatory components, you have a lot of things, and that's how when the helicase job is over, helicase is going to be removed. When the primase job is over, primase is going to be removed. When the SSB is, as the DNA polymerase will be keep moving, right? SSB is also going to be get detached from this double stranded DNA, right? Or the single stranded DNA. So this coordination is missing when you are talking about the in vitro system, and because of that only, it is very difficult to mimic these conditions under the in vitro conditions, right? And otherwise, the data synthesis is a very simple thing, right? You have to do the initiations where you are actually going to prepare the DNA for the synthesis. So in the in vitro system, what we are going to do is, instead of helicase, so in the in vitro, see, this is your natural conditions, right? So if I have to do the initiation, what I'll do is I'll put the helicase, right, in the normal situations. But in the in vitro system, what I'm going to do is I will actually going to heat the sample. I will heat the sample, and if you do the heating, right, the double stranded DNA is going to be broken down into a single stranded DNA, because the heat is also going to be destroy the hydrogen bonding between the nucleotides. And then what I'm going to do is, once the heating is going to generate the single stranded DNA, then what I'm going to do is, instead of using the primase for providing the primer, what I'll do is I will add the primer from outside. Okay, so the primer will go and anneal. Once the primer is going to anneal, right, it is going to sit onto the template. Then I will add the DNA polymerase, right, in and what will happen? The DNA polymerase will go and sit onto this, and then it start synthesizing. So remember that this second strand is anyway not going to be present. So there's no need to add the SSBs. And then they will reach to the end point, and that's how it's actually going to do the terminations. So this is what exactly the people have done when they have discovered the polymerase chain reactions, or the PCR, right? But it took a lot of time, and it took actually significant efforts from the different scientists to achieve this. Very quickly, we'll go through with these efforts. So it started with the discovery of the mechanism of DNA replication itself by the Arthur Kornberg, when he discovered that the DNA polymerase is the enzyme which is responsible for the DNA replication, and then subsequently he discovered the helicases and primase and all that, right? Then in the year of 1976, right, the people have isolated the thermostable DNA polymerase from the thermus equatus, right? Remember that in this particular scheme, you are supposed to heat the DNA after it actually going to form the double stranded DNA, right? And if you heat this particular DNA, right, which is the thermal, the heat labile enzyme, it is actually going to destroy it. So you basically have to keep adding this enzyme after every cycle. And then we also, in the year of 1983, the Kary-Mullis synthesized the DNA oligos, which is for the sickle cell anemia mutations. And then in the year of 1984, the Kary-Mullis and all those people have actually, tested the PCR onto the bigger DNA, right? Earlier they were just using this small DNA, right? But then they started testing this on the genomic DNA. And that's how they actually came up with the idea of the site-specific primers and those, right? And then in the year of 1985, the first patent was filed onto a PCR, and it was applications focusing on the sickle cell anemia. And then in the year of 1985, the people have developed the thermostable DNA polymerase, which actually ease out the problem of the thermal denaturations. And then in the year of 85 to 87, the people have developed the first PCR. So what basically is happening in the PCR is you are actually starting with a double stranded DNA, right? So when you are going to do the denaturation, which means when you are going to heat the sample or heating the sample at 95 degrees Celsius, right? You are actually going to generate the two templates. One template which is of this, the other template is this, right? So this is the number one, this is number two. Now what you're going to do is you're going to add the primers. So the primer is actually going to have the base complementarities, right? So it will go and sit onto the particular template. And then you are actually also going to have the DNA polymerase. So DNA polymerase is going to utilize this double standard initial sequence, and it is actually going to synthesize the DNA. That's how you have generated the templates, right? So you are actually going to have initially you are having the one template molecule. Now you're going to have the two template molecule. One is this, other one is this. Now in the first cycle, right, you are going to have the two template molecules, right? One is this one, other one is this one, right? And if you continue this whole cycle, right, again, right, which means you are going to have another round of denaturations, another round of primer binding annealing, and another round of elongations, then you are going to have the 4 templates, right? So you're going to have the 4 templates, then you're going to have 8 templates, then you're going to have 16, and so on, right? So basically you started with 1 template, you are going to have the 2 template, then you're going to have the 4 template, and so on, right? So this will going to be keep going on, right? That means in a very short span of cycles, you can actually be able to have the very large amplification of this single standard or this simple single templates, right? And that is the power of the polymerase chain reactions, right? So if you want to do the polymerase chain reactions, you basically have to do the multiple steps, right? So basically you have the denaturations, you are going to have you're going to have initial denaturation so that you can actually be able to make the complete genomic DNA into a single standard DNA. And then you are going to have so in the stage 1, you are going to have the initial denaturations, and then the stage 2, where you are going to have the repeated cycling of the denaturations, annealing, and the elongations, right? So you will do the denaturation briefly so that the DNA will again going to be denatured, right? And then you are going to have you will actually lower down the temperature, right, at 56 degrees Celsius, or it is annealing temperature is also going to be dependent on the TM of the primers and the templates, right? And then you are going to allow them to anneal for some time. So what will happen is that your DNA is actually going to have the primer is going to anneal, right? So you know that we are going to require the 2 different types of primer, the powered primer and the reverse primer. And then it will bring it to the slightly high temperature, right? And you are actually going to put them into the elongation reactions, right? So this 72 degrees Celsius will initiate the DNA synthesis, and the time for this elongation depends on the length of the DNA. So basically, if you are 100 base pair per minute is the DNA synthesis speed of the DNA polymerase. So suppose I am working with the 2.5 kb DNA, right? Then I have to put 2.5 minutes into the elongation. So that is what is the criteria, right? 2 minutes 30 seconds. Once this is done, then you again will go back to the denaturation stage again, and this cycle will continue for 30 times. Once all these stage 2 is done, then you will enter into the stage 3. You will do the final elongations, and then you are also going to do the final hold onto the 4 degree, right? And after the cycles are completed, the reaction is held at for several minutes to allow the final extension, and then the DNA is going to be fully extended, and then you are going to have the final folds. So how you are going to do all this? You are going to set up the PCR reactions. So basically, you're going to have the initial denaturation, which is for the 95 degrees Celsius for 5 minutes. Then you are going to have the cycles, right, in the stage 2. So you're going to have the denaturations, that is the 95 degrees Celsius for 30 seconds. Then you're going to have the annealing, which is 55 degrees Celsius for 30 seconds. Remember that the 55 degrees Celsius is also not very constant. It depends on the TM of the template and the primer. Okay, so this is not very fixed. Okay, and then you're going to have the extension, which is the 72 degrees Celsius for 45 seconds. So this is also not fixed, right? Timing is also, we have discussed, right? Timing is also going to be decided based on the length of the template, length of the gene, right? So it's going to be 1000 base pair per minute. And then you're going to have a final extension. So this is the part of the stage 2, right? And the 70 degrees for 5 minutes, and then you're going to have the final hold at 4 degrees so that you can use this one. Now, if you want to do the PCR, you're going to have the requirements, right? So basically, you're going to have you basically should know the DNA sequence of the target region, which means you also should know the DNA sequence of the gene which you want to isolate, right? Then you also require because that is the sequence, that is the gene sequence you are going to use for designing the primer. So you are going to require the primers. Primers are typically 20 to 30 base pair in size, and they can be synthesized from the commercial companies. You basically require the 2 different types of primers. You require the forward primer and the reverse primer. Then you also require the thermostable DNA polymerase, such as tag DNA polymerase, which is not inactivated at 95 degrees Celsius. And then you also require a machine which is called as thermal cyclers. I'm sure many of you might have seen the thermal cyclers, right? A typical thermal cycler, which is basically is there in our lab, right? This is an instrument from the applied bioscience system. So what you have is you basically have a well like this, right? So do you see this, all the wells like this, right? And within this, you can actually be able to put the ependophs or the PCR tubes. And on the top, you have this is called as heating block, okay? So, or the thermal block, okay? So this is actually having the thermal block. And the purpose of thermal block is that it is actually going to always be maintained at 100 degrees Celsius, so that the content, whatever you have, should not evaporate. Because remember that if you are heating a sample here with 95 degrees Celsius, right, then the 95 degrees Celsius, the water will evaporate, right? So you can see that if you have some water here, right, and if you are heating this water with the the spirit lamp, right, then what will happen? All the material will go and heat and will evaporate and will sit here, right, instead of here, because you have the low temperature here and you have the high temperature here. So here you have a 95 degrees Celsius, here you have a 37 degrees Celsius, right? So to avoid this, what the thermal block is doing is, thermal block is present on top of this, right, and it is maintaining a temperature of 100 degrees. So basically, you have 95 here, 100 here, so it will not evaporate because there's more heat here, right? And because of that, the all content will remain at the bottom, and it will actually going to perform the different types of reactions, right? And within this block, you basically have the thermostats, and the thermostats are actually going to, or you basically have the cooling and heating systems. And because of that, you can be able to change the different types of temperature. And this is the screen what you can actually be able to use for setting up the different types of conditions. Okay. So the thermal cyclers, now whatever the thermal cycler we are having, the latest ones, right? We are actually using the Peltier based thermal cyclers. So the Peltier based thermal cyclers are actually changing the temperature very rapidly. So they don't require the the ramping up and ramping down, right? That okay, because you don't use the compressors and you don't use the old technology. So Peltier based system is much faster. Now, when you talk about the setting up the PCR reactions, so basically you require the template DNA, right? For example, genomic DNA, right? You can use the genomic DNA, you can use anything else, right? Then you require the primers. So remember, we tried the forward primer and the reverse primer. Then you require the cofactors such as the magnesium chloride. Then you require the DNTP. So these are the building blocks, right? And then you also require the enzyme. What I am not writing is actually the buffer, and you also require the water, right? So remember that the PCR reactions are always being done in a larger volume, right? So mostly we do the 50 microliter reactions. So template DNA, you are going to add as per the source of the template DNA. So if it is a small viral DNA or the short template, for example, when we were doing the COVID testing, right? The you don't need lot of DNA, right? Because the DNA amount is small, right? Or the length is very small, so you can actually just require the 1 picogram to 1 nanogram DNA. But if you are dealing with the genomic DNA, then you actually require more quantity, like the nanogram to microgram. Because of the simple reason that if the DNA amount is more, like if the size of the DNA is more, then the number of copies are going to be less if you put 1 picogram or 1 nanogram, right? So because you whatever the amplification you are going to get, that would be in proportion to not the number of micrograms, it would be in proportion to the number of moles or number of copies of that particular fragment. Then you require to put the primers. So primer would be in the range of 0.1 to 0.5 micromolars. Then you also require to put the magnesium chloride, but because magnesium chloride is a cofactor for the Taq DNA polymerase in the range of 1.5 to 2 millimolar. And then you also require the DNTPs. So 200 micromolar DNTPs, right? So each DNTP, like the ATGC, right, you have to put so that it will be utilized for synthesizing the reactions. And then you also require DNA polymerase. So typically 0.5 to 2 units per 50 microliter reactions. And then you require the Taq DNA polymerase buffers and the water, right? Now talking about the templates, so you basically have the multiple sources for the template. You can have the genomic DNA, you can have the DNA fragments, you can have the plasmids, you can have the viral DNA, or you can actually have the tissue samples, you can actually have the blood smears, you can actually have the the hairs, right? Remember that when I am talking about the hair, I am talking about the hair follicle, because hair does not have any DNA, but hair follicle do, right? And that's how the people are using the hair for doing the PCR, because that actually small amount of the follicle will have the small amount of epithelial cells, and that actually is good enough for doing the PCR. Similarly, people are doing the blood. You can actually use the mucus also. Imagine that the cell is some cells could be present here actually, right? Because you don't need a large quantity of DNA, right? Even if you have a few nanograms of DNA, that should be good enough to give you the amplified product. Then you also require the primers. So primer is a short DNA stretch. You will basically require the forward primer and the reverse primers. Then you require the enzymes. So you basically have the Taq DNA polymerase, which is from the thermus equus, right? And this DNA is stable in the high temperature in the presence of magnesium, and the optimal temperature for the Taq DNA polymerase is the 72 degrees Celsius, and that's why we are doing the extension at 72 degrees Celsius. There are disadvantages of the Taq DNA polymerase, and basically one of the major disadvantages is that it actually misincorporates the bases and into so the rate is that it incorporates one base pair in every 10 to the power 4 nucleotides. For example, if you are amplifying a 400 base pair target, right, which means the 400 base pair gene, then it is actually going to produce the DNA, which will be having the 33% of the molecule, which is actually going to have the mutations. So, and this error distribution is also not, consistent. It's not like it is going to generate the nucleotide 200 wrong. No, it's not like that. It's actually going to have the random mutations. So, because of that, only the tag DNA polymerase is not very common when you are talking about, when you are going to use the tag DNA polymerase for synthesizing or for amplifying the very large templates or large targeted genes actually. Alternatively, you can use actually the PFU DNA, which is having the high, the proofreading activity. So it actually going to have the better amplification. So it will not going to generate the mutations. So this is a simple thing. These are the reagents what you require for setting up the PCR. And once you are going to be done that, right? So you start with the water, right? So the water is going to be added first, followed by the buffer, number 2. Number 3, you are going to add the primers, right? Then you are going to add the magnesium chloride, you're going to add the DNTPs, you're going to add the tags, right? So then your PCR master mix is ready. And then what you're going to do is you're going to put that along with the genomic DNA. So you're going to add the genomic DNA at the end. And that's how you are going to have the PCR reactions ready. Once this is ready, then you can actually be able to set up the PCR cycles. So in the stage 1, you are going to put the initial denaturations, and then in the stage 2, you are going to put the denaturations, annealing, and the extensions, and you can do this for 25 to 30 cycles. And in the stage 3, it is going to have the final extensions and the final hold. When you're doing this, you have a lot of options. You can actually be able to do the hot start, right? What is mean by the hot start is that you don't put the reactions when, see, what will happen is that, your machine would be at 37 degrees Celsius, right? The moment you turn on the machine, right? And then ultimately it will reach to 95 degrees Celsius, right? So during this ramping, right, it will actually allow your primer. So suppose you are having the append off, right? And if you put the append off into a machine which is cold, right, which is at 37 degrees Celsius, then your mixture is going to experience the temperature starting from 37 to 95. And this is good enough for the primers to go and bind to some of the, your templates. And because the primers are binding before the template is completely open or the single standard, it may actually, there is a possibility that it may actually bind to the wrong places, right? So to avoid this, what people do is people do the hot start, right? Which means they actually bring the reactions at 95 degrees Celsius. They don't add the primers in the reactions. Okay, so they don't add the primers. Then what they do is accept the primers, right? So from this mixture, they don't put the primers, right? Accept the primers. They bring everything into the machine. Let the machine, bring the temperature at 95 degrees Celsius, and then they take out the tube and add the primers. Okay, and then they initiate the reaction. So that actually reduces the non-specific amplification or the non-specific annealing of the primers to the incomplete templates. This is one of the quick method what people do for correcting the non-specific amplifications. So primers are very, very important. The purity and the quality of the primer is only what is going to decide what will be the amplification you are going to get. Apart from the primers, you're going to have the enzymes, you're going to have the additives. So you can actually be able to do the optimizations. If there will be no product which is going to be formed, you can actually be able to check the the quality of the DNA, which means the genomic DNA. You can actually be able to reduce the annealing temperatures. You can increase the magnesium. So these are some of the things what you can actually be able to do for optimizations. So basically, you can reduce the annealing temperature so that the more and more primers should go and anneal, and then you can actually be able to do the fine tuning. You can use the thermostable enzymes. You can actually be able to. So if any of these things doesn't work, right, you can actually have to design the new primer, because your primer could be a source of the problems. So if this is the when the product is not formed, this is when you are actually getting the huge amount of products, right? So in those cases, you can actually increase the annealing temperature or reduce the magnesium concentration or the reduce number of cycles or try the different types of enzymes. So ultimately, what you're going to do after the post PCR, you're going to get the amplified product, right? And this is the amplified product what you are going to get. And these are the primers which are not been annealed to the template, right? And what is the advantage of the PCR? The advantage of the PCR is that it is very fast, it is easy to use, it is very sensitive, and it is also very robust, which means it is not going to give you the non-specific products. What are the limitations? One of the fourth most limitation of the PCR is that it actually requires the DNA sequence, which because it is so until you don't have the genomic sequence known, you cannot be able to do the PCR. Okay, so do you have any question now? So I think we already reached to a point where we can actually be able to, ask you to.
So thank you, sir. If anybody has any question, you may raise your hands. And Manushi Mondal has put here a question. What will happen if the template DNA contains a mutation?
Uh, can you repeat one more time?
What will happen if the template DNA contains a mutation? Obviously, mutation will.
Okay, if the template DNA has a mutation, it is actually going to propagate into the subsequent cycle. So, for example, you started with the template one, right? And this is does not have any mutations. Now, when you do the first cycle of the PCR, you have generated a mutant, right? Now, this mutant will go into the subsequent generations, and it could in the if you're not doing the PCR properly, it could be a mutation on this site, it could be a mutation in the second site, right? So mutation is actually going to affect multiple way. It is actually going to change the amino acids, what is being responsible for synthesis, right? So for example, if there will be a and if it got changed to T, right, then there could be a significant mutations, and it could actually be able to alter the final amino acid, what is going to be code for this template versus this template. So it may ultimately going to change the quality of the protein.
Exactly. She has another question. How complementary base pairing ensures accuracy while copying template DNA?
So complementarity is ensuring because A is always making a pair with T and G is always making a pair with C, right? So if so, they will actually going to follow this rule only, right? So it's not like the other combination is possible, and because of that only the specificity in the things are going to be there, right? So it actually if suppose imagine a situation that if I add like A, T, and I don't add the C, and I add only G nucleotides, right? So remember that this reaction, right? Uh, right. So in this particular, PCR reactions, you are adding the DNTPs, right? If I add only A, T, and C, and if I don't add the G, for example, then your PCR will not work because the G is going to be missing, and wherever it will find the C, it's actually going to require a G to incorporate, and in that case, it is not going to work. So this complementarity provides the specificity into the system, and that's how it is actually giving you the reliable amplifications.
Exactly. Thank you, sir.
Okay, thank
Another
you, sir.
another question from Orijit Dash. How to design probe for unknown mutated sequence?
How to design the probe for
For unknown mutated sequence.
unknown mutated sequence? No, you cannot design the probe for unknown sequence. You actually require the sequence for designing the probe, because
Exactly.
primary requirement of the probe is that you have to have some way to be able to perform the either the reverse transcriptase reactions or the DNA polymerase reaction.
Exactly.
You
Actually,
cannot do that until you don't know the sequences.
we have to construct probe accordingly what we need actually. No.
The probe is required, but until you don't have the small DNA fragment which you are going to use for a probe, that sequence information is not known, then you cannot prepare the
Exactly.
radioactive.
Exactly. Thank you, sir. Another question from Tonri Marshall. So what do we do about primer selection when an unknown DNA sample of small amount needed to be amplified in case of forensic analysis?
Uh, when she says unknown means the person is not known, but when you are talking about the unknown means the DNA sequence of that particular sequence is known, right? So, for example, if we are trying to investigate a crime site, for example, right? So we don't know whether this blood sample belongs to X person or Y person or Z person, right? But if we go by the criminal cases, right, we basically know who are going to be suspect, right? So if the A, B, C, D is suspect, we can actually be able to do the amplification, like suppose whatever the DNA we got at the crime site, right? We can actually be able to see whether that DNA is matching to A or B or C or D, right? So nothing is unknown, right? You cannot just simply have a DNA and you can be able to know which to whom this particular blood belongs, right? So that kind of technique is not available, right? You should have some kind of uh, suspects available, right? Then only you can be able to solve these kind of criminal cases using the polymerase chain reactions.
So I think nobody has any more question. So
I think
thank
the
you.
Mansi was trying to ask me something. I don't know what she was.
Someone put raise hand.
No, she I think she unmute and say something, but I don't know
Oh,
whether she is still available or.
okay. So thank you, sir, for
Yeah.
sharing your valuable time with us and give us interesting insights regarding all these molecular reactions, PCR reactions. And we now it's time for lunch break,
Okay.
and we'll be right in this. um Here we have 2 p.m. From 2 p.m., the next session will start, and sir will demonstrate the practical aspects of this gene cloning, etc.
Hmm.
So please, you take your lunch and come sharp within 2 p.m. Thank you,
Okay.
sir.
Yeah. Sure. Sure.
Okay, thank you, sir.
Okay. Bye bye. Bye bye, everybody. Okay.
Thank you. Bye.